Bhatnagar has a vast experience of 40 years in teaching and research. He serves as a Dean, College of Biotechnology in SVP University of Agriculture and Technology, Meerut. He worked on several administrative positions like Chief Personnel Officer, Nodal Officer, College of Basic Science and Humanities, Chief Warden, Chairman, Screening Committees for CAS, Chairman, Discipline Committee, Director, Training and Placements, besides officiating as a registrar several times in the same university. Professor Bhatnagar established internationally recognized Society for the Plant Research in the year 1988. Presently, Professor Bhatnagar sir is the editor-in-chief of VEGETOS. He is fellow of the Botanical Society of India, member of Phy- Phy- member of Psychological Society of France, fellow of Linnean Society of London, and honorary life member PGPR, Urban, USA. He has been honored by the most prestigious VASVIK award in 2010. Dr. Professor Bhatnagar sir established and started state first department of biotechnology at Bareilly College, Bareilly in 1994. Professor Bhatnagar sir was elected as a member of National Assessment and Accreditation Council, that's NAC, as an autonomous institution of University Grants Commission. He assessed prestigious institutions like Annamalai University, Annamalai Nagar, Bhabha Atomic Research Center, BARC, and HBNI, Tata Memorial Center, Gujarat Ayurved University, Jamnagar, Veer Narmada South Gujarat University, Surat, People's University, Bhopal, and SS Constitute College in Ranchi. He is on expert panel of Agricultural Scientist Recruitment Board in New Delhi. Dr. Bhatnagar is the life member and officer bearer advisor of many societies. Professor Bhatnagar had been instrumental in accreditation of SVP University of Agricultural and Technology, Meerut, by ICAR peer review team and being as a coordinator of ICAR impact assessment during 2017. He is the expert of Project Evaluation Council CCRAS, Ministry of Ayush, Government of India, Uttarakhand Council of Biotechnology, Government of Uttarakhand, and nominated member of Research Appraisal Committee and Research Council for the CSIR NLM Jamshedpur. He was invited to deliver the keynote address in an international conference held in Rome, Italy, an international conference on the industrial biotechnology, Copenhagen, Denmark, which he delivered this, as a keynote speaker by online mode. Professor Bhatnagar produced 16 PhD students, one research associate, and large number of MTech students, beside publishing more than 100 research papers in the reputed journals, peer-reviewed journals, and many other books also. He visited nearly 10 countries, which include France, United Kingdom, the Netherlands, Greece, Ireland, Belgium, Oxford, Macedonia, Indonesia, etc., under various academic programs like INSA, COSTED Fellow, and Indo-Greek Fellow, and as a sponsored visiting scientist. With such a, this is only the brief biodata I have read now. Yes, sir. Welcome you, sir. With this such a brief biodata of this impressive personality, With his contribution, devotion, and achievements, we will start the session. Welcome, sir. Thank you.
Thank you very much for presenting an elaborated biodata. I'm not sure whether I deserve for this much introduction or not. But as I told that being a teacher for past more than 40 years, we are still a student and keep learning so many things from our younger generation. And the association with younger generation keep me agile and active because I feel myself motivated by the upcoming scientists who are having lot of challenges in their career. Such challenges were there during initial stages of our career, but today it is highly competitive world and the younger generation, they are fighting to establish themselves in the respective positions. It keeps me motivated, particularly for connecting myself and for advising, for guiding the younger scientists and younger generations not to give up anything, not to give up in their career, and failure is the first step of your success. If somebody fails at particular place or in a particular objective, we have seen that many younger people, they get disheartened, they undergo depression, or they feel to give up the object. This is not so. If we have achieved this objective, we have achieved this goal, it does not mean that we have not came across any failure. In the initial stages, we might have encountered failure at various stages. And by overcoming those failures, everybody reaches to a definite height. So all the successful people, they should be kept as an object by the younger people, and simultaneously they should be prepared to understand that these people, they have faced all atrocities, all shortcomings, all hurdles in their career, and by overcoming them, they reach to this strata. So this should be the objective of every young people who are sitting over here and listening to this very important national conference. At the outset, let me congratulate the organizers of this national conference on frontiers in life sciences and material sciences at the AI-IPR interface, being organized in a hybrid mode. I would have loved to be there in an offline interaction, but due to over-occupied schedule, I could not make it up, and I preferred to be in an online mode and address the august audience. Now, before we start talking about my presentation, I would like to emphasize that this presentation has been prepared to enlighten the scientist in the field of research publication. And in this presentation, I have tried to take back all the younger scientists to the past. The researches in 1970s or even earlier to that time, they were different. And in the modern time of technological advancement, we are having large number of tools and it is making our task easy. But during our time, when we obtained PhD long back in 1980 from Banaras Hindu University, there were so many challenges. The technologies were not so advanced, and we were always moving from here and there in search of various literatures, in finding out the literature, in identifying the area on which we have to work. But the present generation is having a lot of exposure, and I urge that all of them should make use of the modern technology in a better way, in a positive direction. Since I had been handling the society for last 37 years, and the journal Vegetos, which was spelled by Madam Silpa as Vegetos. In fact, it is Vegetos, which relates to the vegetation, to the agricultural area, to the plant sciences, plant biotechnology, and agriculture biotechnology. So it was established in 1988, and running a journal for 37 years is a big challenge. So with the help of younger fellows, my associates, we had been able to pull it on up to 37 years. And now I wish that the younger generation should come forward and they should carry forward this very important task, which has now become a global journal and is being loved by the plant biotechnologists world over. So let me share the slide because we are running short of time and we need to have other lectures also. Is it visible?
Yes, sir.
Yes, sir. Yes.
Okay, wonderful. Thank you very much. So for this lecture, I selected the topic of AI-driven research vis-a-vis publication ethics. Now, the reason behind selecting this very important topic was that we keep on organizing the conferences. We keep on publishing our data in journals. We go for patenting. We go for the publication in various other magazines. But there is a publication ethics. And if we are following publication ethics while making a publication, either in a journal or a magazine or going for patent filing, we need to be very focused. We need to be aware of the publication ethics, because of which I selected this topic, that in the present time, most of the scientists, they are using the modern tools, ChatGPT and AI, and the research has become AI-driven. But while utilizing these technologies, we should be very clear about the publication ethics. Now, I would like to take you back to the time when we started using our own brain, researches across the time. In the initial stages, there was no artificial intelligence, no ChatGPT, no internet, no communication media, except our brain, which was working like a computer. And still, it is a computer. Because we remember the things which are of our childhood, which are 70 years old or 80 years old, or whatever our grandfather, great-grandfather, great-grandmother, they told us, they remain in our memory under subconsciousness. So you can compare your own brain with the modern computer. So that was called as the real intelligence. And you can see in this picture, there is a vernier caliper, which everybody knows. It is trying to expand our understanding, trying to expand our intelligentsia, trying to expand our intelligence and IQ. So that was the idea, or that was the real research which was going on in the earlier time. There was no option except using our own brain, and there was no option except expanding our brain, our understanding about the subject. Whatever old theories you study, maybe the Mendel's laws, maybe the Darwin's theory, maybe Newton's theory, or anything, they were developed by those persons using their own brain, not by any technological advancement. Now, in the earlier time, we used to have two very important documents. My motive is to apprise the younger scientist about biological abstract and current contents. These were the two important databases when a person starts doing research. Their supervisor, they used to ask them that visit some library, good library, where you can see biological abstract or BIOSes or current content, for identifying, for finding out various research articles published by various scientists the world over. As I told that there was no internet, there was no websites, nothing. So it was only the hard copies of biological abstract and current content. And at that time, we used to identify the papers, used to prepare a small card, as you can see at the bottom of the slide. And on these cards, we used to send the reprint request through post, and the reprints were sent by the author. Nowadays, the reprints or the hard copies of reprints and journals is very uncommon. The people, they are going to the search engine like Google or any other search engine, and they are finding out the paper, downloading it from the website. So at that time, there was no such facility, and we had to depend on these two important databases to find out the real researches. Now, with this slide, I wish to emphasize that quality research was an inbuilt passion. Only those persons who are going for research who are really having a passion to work, having a passion to research, to do something new. Nowadays, we call it as a profession because we joined teaching by our passion, and nowadays we are joining teaching or research by considering it as a profession. Because we are having lucrative salaries, we are having lucrative, scholarships. So we are attracted by a good financial standing. If we are going for research or we are going for teaching, somewhere it is our motive that we should have a good salary, we should be established in our career. But during earlier days, it was not so. I'm not saying that the quality of research has gone down, but at that time, the priority was the quality research, and only those persons were going for research or teaching who are really having a passion for it. And now passion and profession, they have merged together. You can see that that was my first visit abroad, and it was in the laboratory of Professor Micheline Gourlasquin in Angers, France, near Paris. So we used to work over there from 9 o'clock to 5 o'clock, and we were working on Giemsa-C bending, the kinetochore differentiation in lower plants, particularly in charophytes. So kinetochore, to which we call as heterochromatin, was an important feature, which was a deciding factor for identifying the phylogenetic alliances. And with the help of these researches, we could establish that the carrels, Chara, Nightella, Tolipella, these type of algal plants, macrophytes, they are the immediate ancestors of land plants or the angiosperms. So that was our first study on Giemsa sea bending the world over, and we published quite a good number of papers on Giemsa sea bending. Now you can see a lady in between, and she is Professor Micheline Gurleskin from University Cathode in Angers. She visited my institute where we work, and when she visited, we were having a small microscope and a handmade camera, bellow type camera, with the help of which I showed that how the chromosomes can be separated, how they can be stained, and how their picture can be taken with the help of a handmade camera. So she was so captivated by this type of research that she made a small comment that I am very much impressed by the Indian scientists who are doing a good quality of research even without having the basic facilities. And we in France, even after having good equipment, we are even not able to take any photograph of chromosome as on today. So during 1984, when she visited, she was not using any chromosome photograph while we published many papers with chromosome photographs. So this was the difference between the researches in developed countries and the researches in our underdeveloped country, where we were doing a good amount of research without basic facilities. Now you can see these type of cameras. This picture I deliberately took from a museum in Indian Veterinary Research Institute, where one of my old friend, Professor Ramesh Somvanshi, he showed me different types of microscopes which were being used by the earlier researchers, and we also used it during our research and during the research done by many of our students. So these were small binoculars which we used to take to the field, and we used to take the pictures or could identify the plants in the field itself and take photographs with the help of these trinocular microscopes. So, of course, few microscopes are still available in various laboratories, but their advanced version has been introduced. So these were the basic facilities which we were having during last years. Now you can see here the picture of two very important legends who had been my teacher and my grand teacher. On the top, you can see Professor M.B.E. Godward from Queen Mary College, University of London. She was the founder of many algal schools, including at BHU, at Hyderabad, at Rajasthan, and in so many places in India. All the algal cytogeneticists, they were the student of Professor M.B. Godward. She was known to introduce Godward iron alum acetocarbene technique for staining chromosomes. Many of younger fellows might be staining chromosomes by using acetocarbene or acetoarsine. So she was a lady who introduced this technique for the first time and is known as Godward's iron alum acetocarbene technique. She introduced it in 1948. Now, these are my alma maters. You can see down Professor Y.S.R.K. Sharma. He was professor and head at Banaras Hindu University and my supervisor for my PhD. And he was a student of Professor Godward. Now, I wish to emphasize that during that time, since there was no typewriter, no printer, or no advanced technologies like AI to design a letter. So you can see a letter which was written by her in her own handwriting in 1986, September 30, 1986. You can see Professor M.B. Godward, Senior Honorary Research Associate. So at that time, it was indicating the affection and affectionate bonding between the teacher and taught, between the scientist, which is has been diluted nowadays, and we simply send an email and just apprise about our development or about others' development. So this was the tradition during the earlier time of research. Now, these are two important papers which I wish to project. And these two papers were published in Italy and Cytologia from UK. These were the papers which were initially published, and at that time there was no impact factor, nothing doing. And the papers were identified based on their quality of research, quality of content present in them, and they were thoroughly reviewed. And these were two papers which I want to emphasize, which helped me in getting my first job as lecturer, to which you call as assistant professor nowadays. So at that time, the researches were measured or were uh, quantified by the help of papers and the quality of research. There was no impact factor, no citation index, no CITE score, nothing doing. So these were the methods which we had been using in the earlier time of research. Here is an incubation of an embryo from one egg to another, or from one cell to another, and these type of demonstrations we used to give to our students and the faculties so that they were able to perform the researches. There was no virtual experiment. I mean, there was no internet, as I told, so all the experiments were being done by hand, and it helped all the scientists and students to develop a sort of practice, to develop practice in performing the experiments. I would like to emphasize that when I started Department of Biotechnology at Bareilly, all the contents, all the reagents, which we normally called as EDTA or something, which are available in a ready-mode manner by various multinational companies, we trained our students to prepare all these reagents in the laboratory themselves so that they were able to prepare these reagents by maintaining their pH, and they were able to identify that if we are preparing this reagent, then what shall be the outcome, how the DNA can be isolated from a plant material or from an animal material, and how the reagents work. But nowadays, we have got the biotechnological kits developed by different multinational companies. So this was a difference, that we used to work ourselves, and we used to identify that what type of reagent shall be used. These were the conferences which we started organizing in 1994. You can see some legends over here. Professor K.S. Bhargav, who is a legend in microbiology and in aquatic mycology, he was our first president of Society for Plant Research. Next to him, you can see Professor P.K. Gupta, whose books in biotechnology, cell biology, molecular biology are quite popular. He was the guest of honor in our conference, which was organized in 1984. And in the backdrop, near the backdrop, you can see Professor B.M. Johri. He was a legend of Delhi University, and he wrote a large number of books, and he was the chief guest during our conference. So in 1994, we were able to invite such internationally renowned personalities and they were pleased to join us at that time. And in the slide, in the pictures below, you can see that we organized a very important international conference, TASAD India, in 2017, where the scientists from and diplomats from 27 countries from all over the world, they attended this conference in Delhi, in NAS complex. It was organized by the Society for Plant Research. These are few regions I wished to project this slide so that the younger scientists may be able to understand, to know about them. On the left-hand side, he was the director, College Development Council, Professor BBS Rajada, then Professor S.K. Sapori, Professor M.P. Yadav, Professor R.K. Gupta, Professor T.S. Kahlon from California, myself, Professor B.P. Singh, Head of Botany Department at Delhi University, Professor Paramjeet Khurana, Professor Manju Sharma, who was the Director of Department of Biotechnology, Professor R.B. Singh, who is a chairperson and chancellor of many agriculture universities, then Professor Anupam Verma. They were all present during our conference. So during that time, we were able to invite the legends in plant sciences and in agriculture, and all of them, they very gladly represented their institutes and guided our scientists. Now, I had an opportunity to organize a conference at National University of Ireland in Galway, Ireland, during 2008, and where I met the General Secretary of International Phycological Society. So this is just for information. Now, this is my initial institute where I worked since 1977 to 2007. And from there, we established the Society for Plant Research and the journal Vegetos in 1988. And it is now 190 years old, a biggest institution of northern part of India, Bareilly College, to which we call as. And this was another slide, another photograph which you can see that include Professor V.M. Katoch, who is had been the director and Director General of JALMA, or who was the Director General of ICMR, Indian Council of Medical Research. And then next to him is the Vice Chancellor of Amity University, Rajasthan. Then Professor Rakesh Bhatnagar, who was the Vice Chancellor of Humayun University and Banaras Hindu University. So I was fortunate to chair the session where the lectures by Professor V.M. Katoch and Professor Rakesh Bhatnagar along with Professor H.N. Verma were there. So now let us go for the impact factor and research ethics, which was introduced long back in 1975. Till then, nobody was aware of that what impact factor is. And this term, journal citation report, was introduced by Eugene Garfield in 1975, as you can see. And this impact factor is the average number of citation. If we publish a paper, how many scientists they are studying that paper and are citing those references in their own paper, that was called as the impact factor. And the calculation was done by various methods. The citation in last 2 years and the number of articles present over there. The purpose of this impact factor was to identify the selected journals, the important journals, and the institutions where quality research is being done. So since 1975, it was not in force, and the people who published the papers from 1975 or earlier to that, or even in the next 10 years, they were not aware of the impact factor because it was not popular. It was simply introduced at certain institutes and certain levels. The limitation of impact factor was that it is an average, not an indicator of individual article quality. Impact factor does not mean that the article was of good quality, because the quality was judged by the experts And even those papers which were having no impact factor, they were considered as the quality papers which were published in Nature and other journals of high global recognition. And even today, I used to attend or invited for various meetings by Springer Nature. So we are emphasizing that rather than impact factor, we should focus on the real quality of research present over there or published in the journals. Now, Scopus came into existence. And Scopus was a database which provided a global vicinity having citation metrics and a broad coverage of the research articles. Because whatever research we are publishing that need to be disseminated, that need to reach to various parts of the world so that the people must understand that what type of research is being carried out. So if you choose a Scopus Index journal, what you have in your mind that it has a broader scientific exploration because the citation metrics across the field is very important and that is measured by Scopus. Global recognition is given to the journals and we identify that what paper has been done out of funding from different outsourcing agencies. The paper, they receive site score and their impact evaluation is judged by Scopus that what is the impact of this particular research on the society, on the scientists, and how many scientists are implementing this research in their own areas. And we have the broader coverage. While publishing a paper, there are research ethics and it involves honesty. I need to emphasize that the real research is the other name of honesty. No matter whether we are publishing a paper worth 1 page or 2 page or 3 pages, if it is having the real quality of research, which you can see that many papers which were published long back in 1970s or even in 1960s, they were very small, very little, 1 or 2 pages, and they are still referred and cited by many scientists, rather than the papers which are of 20 pages or 30 pages. Unnecessary elaboration is there, unnecessary elaboration of sentences and material just to expand that the paper should be thick. The thickness of paper does not matter, but the real quality of the research is quite important. Their research design is important, data management is important. Then the protection of human and animal subjects, which should avoid the conflict of interest. Conflict of interest means when we have carried out a research and somebody else has published it, maybe he must be our collaborator and he has published it without acknowledging, without involving the real conceptualizing agent or conceptualizing scientist, and then it is a conflict of interest. If some type or same type of researches are being carried out at 2 or 3 places and somebody is borrowing data from others' research and is publishing, that is very unethical and that is taken into account by the ethical committees.
सर
Now,
ये राघवेंद्र सिंह का है ही नहीं क्या?
pardon? Do you want to have any question?
हाँ.
Kindly keep your mic mute,
Okay.
Dr. Banginwar.
Yes, sir. 1
Please
minute.
keep your mic mute here, kindly. This was the statistics for Vegetos, just for a reference that the Vegetos is being served by so many countries and there are many more which I could not enlist over here. Now the Vegetos as on today is having the site score 2.5, which is a good score. It started from 0.5 and then it increases by 0.1, 0.2, 0.3 like that. And now it is 2.5 and its Q3 is 0.25. Now you can see that Vegetos received Growth Impact Award in 2025 by the director of Springer Nature in an event at Delhi, and it was a very prestigious award which was conferred to Vegetos for establishing or for leaving a good impact globally on the scientific community. This slide indicates the authorities, the vice presidents of Springer Nature, and our executive publisher. In the front, you can see Dr. Mamta Kapla, who had been instrumental in helping Vegetos step by step. And this is the award which we received during that conference, during that workshop. It was a big achievement to the journal globally. And now you can see that we had been, I was invited to various workshops. It is called a Journal Development Symposium, and in 2024, I was invited to chair a session, and you can see various vice presidents and authorities of Springer Nature which were together to witness this event. This was the Journal Development Symposium, and you can see Venkatesh Sarasiddhi, Marika Kambin, Deborah Sweet, and myself who were presenting the session. Now, this is very important slide for the younger scientist and the scientist of plant sciences and biotechnology. This is called as COPE. COPE, you all understand, it is Committee on Publication Ethics. Vegetos is a member of COPE. There are very few journals who are the member of COPE. The head office of COPE is in UK. And with the help of this committee, COPE committee, we identify various types of complaints which will arrive to us, like somebody has copied the data, somebody has copy-pasted the details, or has undergone plagiarism, has omitted the real scientist and has not included him as an author. He has repeated the publication, has already published the paper earlier and has now published it again by his own name or by adding few other authors. All type of ethical practices. They come to our committee and we finally decide that what action should be taken on this particular paper. So this is a very important ethical committee where publication ethics is followed. And you can see that the motive of this committee is to have good publication and to handle the research misconduct. At certain universities or institutes, you will find that the misconduct is happening either at the level of supervisor or at the level of a student. Some of the students, they work in a project for 3 years and they publish the data of that project independently without involving the principal investigator. That comes under research misconduct. Likewise, the scientist who took work from a research scholar or from a research associate, and after his leaving the project, they publish the data without involving the real research associate into the paper as an author. So all these research misconducts are happening, and we urge through COPE that there are several guidelines, definite guidelines, where the institutes, they should own the responsibility of identifying such research misconduct at their institute and should inform the committee that this paper has been borrowed from somewhere, that has been copied, or the research has been stolen by the researcher or by the communicating author. And these are the guidelines. We are not going in details of it. Just reading out the title, transparency and accountability is the main reason. Editorial independence, editor can take the independent decision. Handling of misconduct, peer review, authorship, different types of disputes appear among author that they have not included my name. However, I was the part of the team. So conflict of interest and competing interest. Competing interest is about the authors who are on the editorial board of the society or of the journal. And if they are publishing a paper, they should declare that this paper has not been processed by me, or it has been processed independently without bringing it to my knowledge. So I do not have any interference in the peer review of this. That is called as competing interest. And then data integrity. If some data is there, then we take a declaration that the data is authentic and its data availability can be considered by anyone. Then COPE's retraction guideline. This is very important because many papers are being published by the journals and the authors, they have copied it from somewhere and some other author has reported it to COPE that this data belongs to me and I worked under a project or under the financial assistance from somewhere, and this gentleman has published my data in your journal without bringing it to my notice or without seeking no objection from me, or this photograph has been used by them. So that comes under this guideline, and the paper is retracted. And many journals from various international publishers are being retracted in the recent years, which you might be witnessing. But we are fortunate that for last 37 years, Vegetos has not encountered any retraction so far. So we are trying to maintain our integrity and the real quality of research. Now, beside this, Vegetos is also a member of DORA. DORA is called a Declaration on Research Assessment. We try to identify the research assessment by putting them on the screening or by screening their contents. And there is also a committee, and Vegetos is also a part of DORA. It is an authorized signatory of DORA through which we make the assessment, research assessment. It was established by American Society of Cell Biology in San Francisco. Now, the present scenario, we are reaching to the end of this presentation. These were the types of research where we establish integrity, where we establish quality of research, and where we have the impact factor, site score, and different types of quality. We try to maintain quality of our research world over. But nowadays, there are many journals which are developing a practice of publishing the paper in a quick publication mode. Like they will publish the papers in 2 days. They will have very quick processing. They will take money from the authors and will publish it as soon as possible. They are neither having the Scopus index, they are not being indexed in NAS, or they are not having any other inclusion in the database, and their global visibility is almost zero. So I would urge the authors not to be trapped by such journals which are having a very quick processing system or quick publishing system, because for want of promotion or for submitting their PhD thesis, some of the research guides and the students and scientists, they are being trapped by such publications. So that is not going to help them in their real life. So the second thing is that with the introduction of artificial intelligence, why we are focusing on artificial intelligence? Why we have started losing the faith on our own intelligence? Our intelligence, the computer which is built in our brain, is the most important intelligence, or it is the most important tool which should be used for research. Artificial intelligence is to help us. It is a very important tool which can help you in identifying various researches being carried out globally, because whatever data you are providing to the cloud mode, that is taken up by artificial intelligence. So whatever data you are feeding, I came across one important information that in Vegetos it was showing an editor-in-chief of somebody else. The name of somebody else was the editor-in-chief of Vegetos. Then I made a correction, and then AI thanked me that thank you, Professor Bhatnagar, for correcting our data. So whatever data we are feeding, that is being taken up by artificial intelligence and is providing to you on your demand. So don't trust blindly on artificial intelligence. It is very helpful in refining the language, refining the grammatical errors, and refining your papers. But if you are trying to get the data, because when we send the papers for review, that those papers sometimes go to the air or are preserved or are reserved in the cloud, and they can be utilized by artificial intelligence. And if you are getting data from them, and are manipulating them to prepare a review article or a research article, that is not fair, because every journal has started identifying AI-generated papers. Vegetos is also having a definite cell to identify AI-generated paper, and we immediately identify that how much percentage of the paper is AI-generated, and we reject it right from the very first step. So be aware of the use of artificial intelligence in research, and you should utilize your intelligence and should uh, rectify or should improve, refine your research articles rather than using AI for designing the complete paper. I have seen that the research scholars, they are writing review articles. This is very unfair because the review article is a torch bearer. You have to have the research review article from those scientists who are an authority in the field. But the research scholars and but the young scientists or the upcoming scientists, they do not use their own intelligence. They simply gather the information on a topic from Google or from other search engines and articulate them and submit a review article. Believe me that from Vegetos, 80 to 90% such review articles are rejected at first instance. So we are not entertaining review articles from the career beginners. We are taking research review articles from the authorities in the area of expertise. So with this, I thank you all very much for a patient hearing, and I wish this conference in a virtual mode and the offline mode to be a great success. And I'm sure that the younger scientists who have attended this conference, who are attending this two-day conference, will be benefited by various very important lectures from the authorities and will make use of it in their future career. Thank you very much, and thank the organizers.
Thank you, sir. So, any questions from the participants? The house is open. So, any students or the scholar or academician, sir, my question, sir.
Yes, please.
Sir, in a present scenario, there are n numbers of the publication which is available in the market.
Yeah.
And everyone goes for the publication just for the case.
Yeah.
To get the certain marks.
Exactly.
There are so many the publishers who are been in contact with the academicians. Are you interested in the paper or the research writing or the thesis writing? It is a good scenario for the research persons as an ethical point of view.
Actually, we few years, I think 2-3 years back, we organized two very important webinars, particularly for those who are believing on NAS impact factor and those who are believing in UGC care list, which has now been discontinued. So there were two different webinars. So we unanimously emphasized that the API score which has been introduced for the career advancement schemes or for submission of PhD thesis, that binding of two papers, two published papers, that has potentially harmed research quality. This is what I understand. And most of the persons, they were asking me, sir, you have raised a very burning question, and it is very serious issue because API score. I said that no, API score does not mean that the substandard quality of paper should be entertained. And because of increasing API score, the mushroom growth of false, fictitious, short-lived, and unidentified journals has cropped up. Large number of journals are coming up which are publishing by getting some few papers, few money out of that, and they are publishing the papers, and the API score is being increased. So API score is good if we keep on monitoring the quality of papers which have been included in them. Second thing, the publishers, they should also be identified that which publisher is good enough or which publisher is globally recognized, and what is the mode of publication. Say, for example, I can quote an example of Vegetos itself. It is a Scopus Index journal, as you know, and it is being published by Springer Nature. So we have the publication in hybrid mode. Hybrid mode means those developed countries which have got ONUS, means one nation, one subscription scheme. There are many universities which get funds from the government under ONUS, one nation, one subscription. So if you are publishing a paper and if the government has provided you sufficient fund under ONUS, then you are able to meet out the APC, yani publication charges, applied publication charges, because you have not to pay money from your own pocket. You have been given a grant by the government under ONUS and you can pay it. One thing. In the developed countries or in the underdeveloped countries, or you can say the developing countries like India, Bangladesh, Sri Lanka, Pakistan, and like that, the funds are very scarce. And for that, we are having the hybrid mode of publication in the subscription mode. If the authors are publishing the paper under non-open access, then they are charged not even a single penny by Springer Nature. They are not charged even a single penny. They will publish it under non-open access or subscription mode. For that, what we do, if the author is a life member of Society for Plant Research or Vegetos, we provide them access by giving them the PDF. They can access the entire volume of the journal from our website by using their user ID and password for no cost, for free, lifelong. If they are the life member, if they are the annual member, they can do it for 1 year during their subscription. So that is the facility which Society for Plant Research provide to our life members and to our annual members. So we are in an understanding with Springer Nature that we will provide them PDF if they are our member, only to members, by using their user ID and password. That is for dissemination. You can send it through email to anybody. You can disseminate it yourself to thousands of your contacts. But if you are going for open access under Springer Nature, then you will be applied with the APC, applied publication charges by the Springer, which normally the Indian people, they do not prefer because it is very high, and only those institutes or universities which are covered under ONUS, I told you, one nation, one subscription, only they can get the funds and they can give it to Springer Nature.
Thank you, sir. Thank you. Sir, one more question,
Yeah.
which is in the chat. Use of AI in research writing is ethical or not?
No, it's a debatable question, because if we are, as I told, if we are using artificial intelligence for refining our data, for refining our language, for refining grammatical errors, for writing the cover letter to the editor, then it is fine. Or we are providing data to artificial intelligence and this data is being analyzed using artificial intelligence, it is fine. But if we are borrowing data through artificial intelligence from somebody else's research, then it is unethical and unfair. So such type of issues are coming up before us during COPE meeting, and we address them that AI should not be used for preparing a research article, for preparing a review article, for utilizing the data of others, because that will be caught immediately. Because we have got the software that if AI has provided you the data from other sources, then it can be caught and the paper can be rejected, and not only rejected, the ethical issues or ethical, action can be taken against the author.
Okay. Thank you, sir.
Thank you very much. And I hope I have not consumed much time.
No. So at the last, sir.
Thank you very much.
Out of thanks. Sir explained in a very well manner regarding the AI. At the same time, what is the importance of the impact factor? Many of the terminologies, their own limitations are there regarding the quality research, and she explained the scopus in a well manner. The exploration, global recognition, strong matrices are there, as well as the broad coverage. At the same time, really the research will be the ethical or not. What you have to keep in a mind, the core principles, key areas, purposes, framework. At the same time, sir explain in a very well manner in a short period of a span, that will be the COPE. I really thankful. He spent his own important time and share his ideas on the today's framework that will be the AI driven. Thank you, sir. On the
Thank
behalf
you. Thank you all. Thank you all the
National
audience and
Conference.
wish you good luck.
Thank you, sir.
Thank you.
Yes. Next time we will see in a offline mode.
Sure, sure. I would love to be there.
Yeah.
Thank you.
Thank you, sir,
Thank you.
for your precious time which spent with us.
Thank you.
So now the second technical session as a keynote speaker. Pachori madam, are you joining?
Yes, sir.
Okay. Yeah, madam. So welcome, madam, on the behalf of a national conference, a hybrid mode on frontiers in life sciences and material sciences at the AI IPR interface.
Thank you, sir.
Organized by the 4 different colleges to come together. This is the first time, as per the Dr. A.M. Deshmukh sir. And the today's second resource person as a keynote, Dr. Rachna Pachori Sharma, presently working as the professor and head, Department of Microbiology, UG, PG, and research section, RA College, Washim. I will introduce madam briefly. She's research area, environmental biotechnology, applied microbiology, microbial endophytes, PGPR, biocontrol, biofertilizer and pesticides, soil and agriculture microbiology, nanomicrobial technology, biology, reproductive biology, plant bioactive compounds, MDR in microorganism, applied microbiology. A research theme, the exploring the role of microorganism in initiating the reproductive tract infection with a special reference to the pelvic inflammation diseases in a woman of reproductive age groups. Determination of MDR in clinical isolates and their management and plant extract. Exploration of the plant bioactive components responsible for the antimicrobial activity and special reference to microbial endophytes. Madam have a teaching experience total 20 years, 21 years UG and PG. Madam PhD recognized guide at Sant Gadge Baba, Amravati University, Amravati, Maharashtra. Member Board of Studies in Biochemistry, Microbiology, including Food Science, Sant Gadge Baba, Amravati University, Amravati, since 2017 to till date. Madam, Chairperson in BOS, Biochemistry, Microbiology, including Food Science, 2025 to till date. Madam will be examiner, paper setter, referee at Sant Gadge Baba Amravati University, Amravati, SRTM University, RTM University, postgraduate teacher in microbiology, Sant Gadge Baba Amravati University, Amravati. Recognition as in the pool of expert for academic audit, co-coordinator, clinical laboratory technology certificate, diploma, advanced diploma under the COP scheme of UGC since 2020. Coordinator of PhD Research Center, RA College, Washim, affiliated to Sant Gadge Baba Amravati University, Amravati. Madam honors with a recipient Bharat Gaurav Puraskar and Certificate of Excellence in the field of microbiology. Madam receives the Women Research Award and Certificate of Achievement in International Scientist Award of Engineering, Science and Medicine in 2022. Madam receives the EDU Excellence Award 2022, ED Vishan for outstanding contribution toward the society in the field of education. Madam also receives the Appreciation Award for the explanatory contribution in a National Science Children Literacy Mission for Microbiology Biotechnology 2022 by Microbiology Society, India. Madam also receives the Rashtriya Shiksha Ratna Puraskar 2023 by Real Indo Global Vision Development Gurukul Foundation, Dhule. Receive the Bharat Ratna Dr. APJ Abdul Kalam International Honors Award 2023 by Universal Research Ground, Nanded. Madam publishes so many research papers, PhD guided, ongoing 5 students, minor research project completed, total publication of 56, national and international in peer review. Madam also submitted the sequences to the NCBI Gene Bank and published near about more than 10 books and chapters. With this short biodata of Dr. Rachna Pachori Madam, RA College, Washim. Over to Pachori Madam.
Thank you very much, sir, for giving the introduction. So should I share my presentation?
Yeah, definitely, madam.
Sir, I am unable to share the screen.
No, you are as a yes.
I think
You should proceed, ma'am. Already you are a co-host.
yes, sir, but
Bhatnagar sir.
Bhatnagar sir, yes, presentation of Bhatnagar sir is on.
Bhatnagar sir. Are you hear me? Bhatnagar sir.
Bhatnagar sir, can you please
Uh,
end your
Stop
presentation?
sharing.
Stop sharing.
Bhatnagar sir. Madam, please try.
Yes, sir. No, sir. Still I can't share.
Bhatnagar sir.
सर डू वन थिंग, सबको कोच बना है ना, तो उनको रिमूव कर दो।
सर
गेस्ट बना था।
वो co-host रिमूव किया है, प्रोहोक किया है सर.
अच्छा ठीक है।
But still there is an PPT. Bhatnagar sir.
So now is it visible, sir? My
Yes,
presentation?
yes, yes. Yes, yes, yes.
Yes, ma'am.
Okay.
It
Yes,
is
ma'am.
visible.
Okay. Okay. So good afternoon, everyone. As introduced by Dr. Banginwar sir, I am Professor Dr. Rachna Pachori Sharma, and presently I am working in RA College, Washim. So today I am discussing on the topic traditional insights for modern sustainability. Now, basically, why I had choose this theme, traditional insights for modern sustainability, because we all know that our India is having a rich treasure of knowledge, and in the past we are using different type of natural remedies for different type of problems, and slowly we shifted over different type of new chemicals or different type of you can say uh, chemical fertilizers. So for maintaining the sustainability in the modern period, now we have to revert back to the traditional knowledge. So in my presentation, I will discuss about the different themes regarding with respect to microbiology. So microbial sustainable hub. In my presentation, I will discuss about the agriculture and soil microbiome restoration, health and wellness, then gut microbiome, food preservation. Just a minute. What happened? Food preservation and microbial sustainability, climate change mitigation, environmental bioremediation and ecological balance, microbial bioenergy and renewable systems, artificial intelligence in microbial sustainability. Now, basically, what traditional insights for modern sustainability means. So here we are integrating the ancient eco-friendly wisdom with contemporary technology to create a regenerative future. We have to generate the future which is regenerative. So in this topic, the key features are circular economy and resource management. Because in the traditional history, the waste is treated as a resource. So first we will discuss about the agriculture and soil microbiome restoration. So agriculture in distress. What are the present situation in agriculture? We see that barren and desertified land is increasing. Soil infertility and micronutrient are depleted. Chemical fertilizer dependency is more. Resistant plant pathogens are there. And due to all this, loss of soil microbial diversity is there. So how we can overcome this? So before that, we will focus on nutrient imbalance. Soil nutrient imbalance is disturbed. Somewhere you can see the nutrient depleted soils are present, or somewhere there are nutrient overloaded soils are there. So oversaturation of NPK, it leads to micronutrient depletion. Secondly, bacterial decline is there. PGPR, which we know as rhizobacteria, which is beneficial for the nutrient uptake and root health. So this bacteria is declining in the soil, and fungal collapse is also there. Mycorrhizal colonization is also declining. So what is the mechanism to restore the soil? So basically, for restoring the soil fertility, we can go for nitrogen fixation. So in nitrogen fixation, we can go for different type of cover crops. We can plant the cover crops like legumes, grasses, brassica, which help in nitrogen fixation and which can add organic matter to the soil. We can go for the crop rotation. Then we can also go for phosphate solubilization. Different type of microorganisms, they will secrete the organic acids to unlock the minerals. Then ISR is also there, induced systemic resistance, which will prime the plant immunity. We can boost the plant immunity with the help of induced systemic resistance. And carbon sequestration, microbial humification of organic matter. Now, how we can use the microbiology in traditional methods to overcome this problem? So firstly, we can go for the cow-based bioenhancer. We know that cow is regarding as a god in India. Now, we can obtain different type of products from the cow which are important as a bioenhancer. Like we can go for Panchagavya, Jivamrut, Bijamrut, Sanjivak, and Amrut Pani. Now, presently in our laboratory, we are working on different type of cow-based enhancers, and we got good results. In case of Panchagavya, we had used Panchagavya in different type of soils in the farms, and we got the good results. Now, Panchagavya, it is the mixture of 5 different products obtained from the cow. It involves cow dung, urine, milk, curd, and ghee. Similarly, Jivamrut is also the mixture fermented. It is the mixture of fermented cow dung, urine, jaggery, pulse flour, and soil. So, in this way, cow-based bioenhancer, they will act as a growth promoters, immunity boosters, and soil conditioners. They will condition the soil, and automatically our soil will be enhanced. Now, secondly, we will go for the composting. It increases the microbial biomass and enzymatic activity in the soil, due to which the soil will become soil will get the different type of nutrients. Then we will go for the crop rotation, which is also mentioned in our traditional methods, which is helpful for maintaining the microbial diversity. We will go for the fermented biopreparations, which will introduce active microbial community. In case of fermented biopreparations, we can go for the biochars, which are the enhanced carbon storage and microbial activity enhancers. Then botanical extracts, we can go for that. Botanical extracts, in case of botanical extracts, we can go for the plant derived phytonutrients, which will selectively suppress the pathogenic microbes and enhance crop tolerance to environmental stress. Now, in case of botanical extract, we had also tried different type of projects in our research laboratory, and we obtained good results for enhancing the environmental stress. We work on the drought stress on the plant and applied botanical extracts to overcome the drought stress. So in that project, we got the good results. So we can go for such type of traditional methods. Now, concentrating to the second issue, which is the health and well-being. Now, health and well-being, if you concentrate on this image, the major cause of the health is chronic infections and dysbiosis, which is as a result of alteration or depletion in the gut microbiome. Now, here we can see that dysbiosis means the imbalance of the beneficial microorganisms present in our gut. It is mentioned that our gut, it is having the tremendous activity which is controlling our metabolism, and gut brain axis is there. If our gut is healthy, it will give the signaling, proper signaling to the brain. And if our brain is working properly, our metabolism will be maintained. So if this gut-brain axis is faulty, then it will result in different type of systemic inflammation, autoimmune risk is there, increased inflammatory disorders are there. It is considered that our gut is the second brain of our body. How? Because in our gut, different type of bacteria is present. This bacteria generate neurotransmitters, basically serotonins, and these serotonins give signal to the brain, and this signaling is helpful in different type of conditions like mental health. Our mental health is controlled by the gut microbiome. Our GI tract health is also maintained by the gut microbiome, and different type of neurological disorders like Parkinson disease, Alzheimer's disease, these are also results due to the alteration in the gut microbiome. So what happened in the present day, we are using the antibiotics in overuse, and as a result of tremendous use of antibiotic, the pathogenic microorganisms are destroyed, but as well as the beneficial microorganisms are also destroyed. Due to that, there is a reduced diversity in the gut microbiome, and it leads to different type of infections, different type of biofilm associated wound infections are also there. This wound infections, biofilm associated wound infection, is also as a result of different type of MDR strains. So here we know that WHO has reported that superbugs are the emerging cause of global threat. So these resistant pathogens, it is due to the different mechanisms involved in resistance to the antibiotic. We are using the antibiotics in tremendous amount, and that's why superbugs are creating, and it leads to treatment failure. So this is the present situation related to the health and well-being. Now, what traditionally we can do? So traditional knowledge emphasize on development of plant-based antibiofilm formulation. We can formulate the antibiofilm formulations which are plant-based. So different type of plants are there which are related with the antibiofilm activities. Like we can go for the neem, tulsi, moringa. All these plants, they are helpful for antibiofilm activities. So by deriving the plant-based compounds, we can go for the development of antibiofilm formulations, and these biofilms will be targeted with these compounds. So development of plant-based antibiofilm formulations are recommended. Then designing herbal hydrogels. This is also the emerging field. Hydrogels. Hydrogels are the 3D networks inside which the phytochemicals are trapped. So these hydrogels are the sustained releasing molecules. If we impart the phytocompounds in this hydrogel will release the phytocompounds to the targeted site, and in this way we can overcome with the MDR reduction. So MDR microbe, these are the MDR microbe specific hydrogels. So again, in our laboratory, one of my PhD student is working on the herbal hydrogel formulation, and she has prepared different type of hydrogel like neem hydrogel, turmeric hydrogel, aloe vera hydrogel, and we had tested this hydrogel for the skin pathogens, MDR pathogens, gut MDR pathogens, and we obtained the good results for these hydrogels. And now we are proceeding for this hydrogel towards the patent. Again, we can use phytochemicals as quorum sensing inhibitors. Now we all know that quorum sensing means it is the signaling between the cell to cell communication. Now phytochemicals which are obtained from the plants, these phytochemicals can act as a communication breaker. They can break the communication between the microorganisms. So this phytochemicals, they can act as a natural quorum sensing inhibitors. They disrupt the bacterial communication, and as a result of which the virulence of the bacteria will decrease. So we can decrease the virulence with the help of this natural quorum sensing inhibitors. Then they can also inhibit the biofilm without antibiotic resistance. Now, second approach is that combining botanical extract with controlled microbial therapies. This is again a very good topic for the research. We can combine the botanical extracts which we obtain from the plants with the microbes. Like we can club this with the help of bacteriophages. We can club this with the help of different microbial molecules, and as a result of which we can obtain the dual action approach. Like here we can see botanical plus microbial synergy. So we can go for the research related with the synergistic action between the botanical extracts and the microbes. Then secondly, we can also create microbiome restoring nutraceuticals like probiotics and prebiotic synergy. If we can club probiotics and prebiotic, we can go for the syntropic microbial network. So this syntropic are useful for increasing the beneficial microorganisms in the gut. So our microbial diversity in the gut will increase. And if our gut is healthy, our body will be healthy. So this is the traditional approaches which we impart in modern science. Now next we can go for the gut microbiome health. Now gut microbial problem, which already I had discussed, that reduce microbial diversity in our gut. The microbial diversity is reduced, the beneficial organisms are reduced and harmful organisms are increased. overgrowth of the pathogenic bacteria. Then antibiotic-induced dysbiosis is there. Chronic inflammation and metabolic disorders are there. And as a result of this, leaky gut is there. So how we can overcome from this problem? So we can go for the traditional system which unknowingly support the microbiome diversity. Like in our traditional system, the use of fermented food is mentioned. So introduction of fermented food in our day-to-day life will automatically increase the beneficial microbes, and it will help in the maintenance of microbiome in our gut. Secondly, we can also go for the high fiber plant-based diet. High fiber, we can use the diet which is having the high fiber content. In day-to-day life, what we are consuming? We are consuming carbohydrates more, we are consuming fats more. But instead of carbohydrates and fats, if we will concentrate on the proteins and the fibers, then automatically what will happen? The gut microbiome, it will increase by prebiotic substrates, because this is acting as a prebiotic substrate. So prebiotic, if the prebiotic substrate is increased, automatically the probiotic strains will be increased in our gut, and automatically our immune system will be maintained. Then in the traditional system, herbal decoctions are there. Different type of herbal decoctions are mentioned in traditional methods, like herbal teas are there, cinnamon tea, then lemongrass tea is there. So different type of herbal decoctions, if we are using in our daily routine, so it will selectively enhance the antimicrobial activity in our body, and it will support the immunity, and our immunity will be boosted up. Then seasonal eatings, microbial adaptation support. If we seasonal fruits, in our traditional system it is mentioned कि जिस सीजन में जो फ्रूट आता है वो हमने खाना चाहिए. तो क्यों खाना चाहिए? क्योंकि it is seasonal eating. If this seasonal eating is there, microbial adaptation will support our diet. So traditional diet is having the natural microbiome modulation strategy. So traditional inspire modern microbiome solution. What we can do? So we can develop the standardized probiotic formulation. Like different type of probiotic formulations are there. Again, with respect to this probiotic formulation, one of my PhD student has completed her PhD in the preparation of probioticated aloe vera juice. We had prepared the aloe vera juice which is having the probiotic strain imparted in that. So we are having the advantage of aloe vera as well as the probiotic. Basically, this project, this PhD title, it was focused on the lactose tolerant individuals, because we know that in recent days lactose tolerance is increased in the population. So if the people is lactose tolerance, they can't go for the dairy products. So what we had done, we had isolated the probiotic strains from different dairy products, and these probiotic strains are imparted in the aloe vera juice. So now this aloe vera juice is having the dual benefit. We can get the benefit for the aloe vera as well as we can get the benefit for the probiotic organism. So again, in this PhD research work, we got the good results, and we had done the study with respect to shelf life also. We had also used this preparation on different type of organoleptic properties. So such type of standardized probiotic formulation we can design. Then we can use the prebiotic rich plant extract. Different type of plant extracts are there, like garlic, ginger, turmeric. All these are mentioned in our traditional science, that we can consume this. Different type of spices are there, so we can consume the spices in day-to-day life, so that the prebiotic will increase. Then we can combine phytochemical with microbiome therapy. Phytochemicals which are obtained from the plants. These phytochemicals, if we club this with the microbiome therapy, then ultimately this will give the good results. Then we can design the symbiotic probiotic plus prebiotic system. So here again we can get the dual benefit of probiotic and prebiotic. And nowadays we can also go for the personalized gut microbiome. So again, I like to share something with personalized gut microbiome. It is said that individual to individual, the genome varies, the gut microbiome varies, आप जैसे पैदा हुए हो, आप जिस कल्चर में रहे हो, उस हिसाब से आपका गट माइक्रोबायोम होता है। तो एक मेडिसिन जो एक इंसान के ऊपर काम करेगी वो दूसरे पे नहीं करेगी। तो उसके लिए वी हैव टू गो फॉर द पर्सनलाइज्ड गट माइक्रोबायोम थ्रू सीक्वेंसिंग। तो इंडिविजुअल का पालन पोषण किस बैकग्राउंड में हुआ है, वो कौन से एनवायरनमेंट में सर्वाइव कर रहा है, कहाँ पे काम कर रहा है, उसके आजू बाजू का परिसर क्या है, इसके बेसिस पे हमारा माइक्रोबायोम डिसाइड होगा कि हमने किस तरीके के जो गट माइक्रोबायोम है उनको इंक्रीज करना चाहिए। तो अभी जो एआई का युग आ गया है, इन दिस एआई जनरेशन वी कैन ऑल्सो गो फॉर पर्सनलाइज्ड गट माइक्रोबायोम। सो after that, we can go for the microbiology oriented traditional models for food preservation and storage. Now, in case of food preservation and storage, we know that present day problems are rapid microbial spoilage. फूड जो है इसका माइक्रोबियल स्पॉइलेज हो जाता है, ग्रोथ ऑफ पैथोजेनिक बैक्टीरिया एंड फंजाई, पोस्ट हार्वेस्ट लॉसेस आर देयर, केमिकल प्रिजर्वेटिव डिपेंडेंसी, इन सबको ओवरकम करने के लिए हम लोग केमिकल प्रिजर्वेटिव के ऊपर डिपेंड हो गए हैं। न्यूट्रिएंट डिग्रेडेशन ड्यूरिंग स्टोरेज, जब हम उसको स्टोर करके रखते हैं कोई भी चीज को बहुत दिनों तक, तो उसका न्यूट्रिएंट डिग्रेडेशन होता है। तो ये जो प्रॉब्लम्स है, ये प्रॉब्लम को अगर हमको सॉल्व करना है तो अगेन ट्रेडिशनल वेज आर देयर। अभी ट्रेडिशनल फूड सिस्टम जो है उसमें हम लोगों को बताया गया है अगेन फर्मेंटेशन, जो मैंने पहले भी बताया कि फर्मेंटेशन इज द वेरी गुड प्रोसेस for overcoming many problems related with the food, related with the health. So in fermentation, the beneficial microbes are increased and it will suppress the pathogens. Sun drying, sun की मदद, sun जो है ये तो हमारे लिए नेचुरल फ्री में अवेलेबल है। तो सन ड्राइंग, हमने बहुत सारी चीजें जो है सन ड्राई करके अगर हम प्रिजर्व करते हैं, उसका मॉइस्चर रिडक्शन हो जाएगा, माइक्रोबियल ग्रोथ जो है ये इनिबिट हो जाएगी। देन सॉल्ट एंड नेचुरल एसिड्स, ये जो होते हैं सॉल्ट एंड नेचुरल एसिड ऑस्मोटिक स्ट्रेस ऑन स्पॉइलेज ऑर्गेनिजम्स, इसको ओवरकम करते हैं। अब ये सॉल्ट जो है, सॉल्ट से रिलेटेड भी मैं एक रिसर्च प्रोजेक्ट आपसे डिस्कस करना चाहती हूँ। हम लोगों ने एक रिसर्च प्रोजेक्ट में जो अम्बिलिकल कॉर्ड होता है, जब मदर से बेबी की डिलीवरी होती है उस समय जो प्लासेंटा होता है, वो प्लासेंटा जो है उसको कट करने के बाद हम लोगों ने उसका सैंपल लिया और उसको हमने सॉल्ट से ट्रीट किया। तो वी फाइंड दैट as compared to modern days medicine, this placenta is dried in within 4 days, 4 दिन के अंदर ये सूख गया। तो ऐसी जो जगह है, ऐसे जो हम लोग कहेंगे कि विलेजेस है, रूरल एरियाज है, जहाँ पे मेडिकल फैसिलिटीज बहुत कम है या लोग नहीं जाते, अगर ऐसी जगह पे ये सॉल्ट का यूज किया जाता है फॉर umbilical cord sepsis, to overcome umbilical cord sepsis, तो it is also having a good result. तो salt can be used as a lucrative alternative फॉर अम्बिलिकल कॉर्ड सेप्सिस। तो यहाँ तक का मैकेनिज्म ये सॉल्ट का होता है, क्योंकि इट इज हैविंग द ऑस्मोटिक स्ट्रेस ओवरकम प्रॉपर्टी। देन हर्बल एक्सट्रैक्ट्स, वी नो दैट हर्बल एक्सट्रैक्ट्स दिस आर हैविंग द नेचुरल एंटीमाइक्रोबियल कंपाउंड्स। तो नेचुरल एंटीमाइक्रोबियल कंपाउंड्स जो होते हैं, इसका यूज हम लोग फूड प्रिजर्वेशन के लिए कर सकते हैं। So traditional preservation techniques, it is having the equivalence to ecological microbial regulation without synthetic chemicals. Synthetic chemical का use नहीं करना है and we can go for the traditional preservation techniques. Now we can also focus on microbiology oriented traditional model for sustainable energy production. अभी energy and environmental problem जो अगर हम देखते हैं तो हमको पता चलता है कि fossil fuel dependency is there. We are maximum time we are depend on fossil fuels. As a result of that, rising carbon emissions are there, agriculture waste accumulation is there, industrial pollution is there, and energy insecurity in rural areas. Rural area की बात करते हैं तो energy insecurity का problem आज भी है। Now if we go for the traditional system, so unknowingly traditional system ने हम लोगों को बहुत सारा biological energy cycles दी है, like biomass burning is mentioned in traditional system, algal biofuel system, it is also mentioned in traditional system, microbial fuel cells, we can use the microbes as a fuel cells, and waste to energy bioreactors, we can design the bioreactors which will convert the waste to energy. सो बेसिकली अगर इसका साइंटिफिक इंटरप्रिटेशन देखते हैं तो ट्रेडिशनल एनर्जी प्रैक्टिसेस वेयर अर्ली फॉर्म ऑफ माइक्रोबियल बायोमास कन्वर्शन सिस्टम। ट्रेडिशनल एनर्जी प्रैक्टिसेस में माइक्रोबियल बायोमास का कन्वर्शन किया जाता था। अब इसको हम मॉडर्न माइक्रोबियल एनर्जी सॉल्यूशन से कैसे टाई अप करेंगे? तो वी कैन डिजाइन एनारोबिक डाइजेस्टर्स यूजिंग माइक्रोबियल कंसोर्शिया। माइक्रोबियल कंसोर्शिया हम यूज करेंगे और इसका यूज करेंगे एनारोबिक डाइजेस्टर्स में। वी कैन गो फॉर बायोइथेनॉल प्रोडक्शन वाया माइक्रोबियल फर्मेंटेशन, बायोइथेनॉल प्रोडक्शन करेंगे। अलगल बायोफ्यूल सिस्टम, अभी बहुत सारा रिसर्च जो है अलगी के ऊपर चल रहा है विथ रिस्पेक्ट टू बायोफ्यूल्स. So algal biofuel system, it is again having a good advantage for microbial energy solutions. And waste to energy bioreactors, different type of bioreactors are designed where waste is converted into the energy. Now microbiology, which is related to the environmental sustainability. Now in case of environmental sustainability, present day we see that different type of problems are there, different type of imbalance is present in the environment, like soil and water contamination. अभी पानी और जो सॉइल है इसका कंटेमिनेशन बहुत ज्यादा है. प्लास्टिक एंड बायोमेडिकल वेस्ट एक्यूमुलेशन इज देयर, लॉस ऑफ बायोडाइवर्सिटी, इंडस्ट्रियल केमिकल रन ऑफ इज देयर, रिड्यूस नेचुरल कार्बन सिक्वेस्ट्रेशन इज देयर. सो इनका सबका जो अगर हम कोर इशू देखते हैं तो कोर इशू क्या है? Disruption of natural microbial ecosystem, which leads to pollution persistence and ecological imbalance. Pollution बढ़ रहा है और ecological imbalance disturb हो रहा है. Now, ट्रेडिशनल सिस्टम जो है, ट्रेडिशनल सिस्टम इट वर्क इन हार्मोनी विद द माइक्रोबियल साइकिल्स. जो हम माइक्रोबियल साइकिल्स पढ़ते हैं, नाइट्रोजन साइकिल, कार्बन साइकिल, फॉस्फोरस साइकिल, तो ट्रेडिशनल सिस्टम जो है, ट्रेडिशनल सिस्टम ने ये सब माइक्रोबियल साइकिल के बीच में हार्मोनी मेंटेन की थी. लाइक इन ट्रेडिशनल सिस्टम ऑर्गेनिक वेस्ट कंपोस्टिंग इज मेंशन. पहले के जो लोग थे वो कंपोस्टिंग करते थे जिसमें ऑटोमेटिकली माइक्रोबियल डिकम्पोजिशन होता था. नेचुरल फाइबर यूजेज, मैक्सिमम जो भी मटेरियल होता था वो बायोडिग्रेडेबल होता था. लाइक अगर हम लोग सीधा सीधा देखते हैं तो पुराने जमाने में हम लोग पत्रावड़ी यूज करते थे, कहीं पे भी खाना खाने जाते थे, या फिर बनाना प्लांट की लीव्स यूज करते थे. आज हम लोगों ने उसका कन्वर्शन पेपर में कर दिया. तो पहले के जमाने में बायोडिग्रेडेबल मटेरियल्स आर देयर फॉर डे टू डे लाइफ. देन सेक्रेड ग्रुप प्रिजर्वेशन, बायोडाइवर्सिटी कंजर्वेशन के लिए ग्रुप प्रिजर्वेशन किया जाता था. क्रॉप रेसिड्यू रिसाइकलिंग होता था जिसमें कि क्रॉप का ही रेसिड्यू हम लोग रिसाइकल करते थे फॉर एनहांसमेंट ऑफ द सॉइल फर्टिलिटी. सो साइंटिफिक इंटरप्रिटेशन अगर हम इसका देखते हैं ट्रेडिशनल सिस्टम का, तो हमको पता चलता है कि ट्रेडिशनल इकोलॉजिकल प्रैक्टिसेस दे आर बेसिकली मेंटेनिंग द बैलेंस माइक्रोबियल न्यूट्रिएंट साइकिल एंड मिनिमाइज टॉक्सिक एक्यूमुलेशन. नाउ in modern day what we can do? So in modern technology we can go for the microbial bioremediation of contaminated soil and water. जो भी soil और water का contamination हुआ है उसको माइक्रो ऑर्गेनिज्म की मदद से हम लोग बायोरिमीडिएशन की ओर ले जा सकते हैं और वापस से हम उसको पुराने फॉर्म में लेके आ सकते हैं. Now plastic degrading bacteria and enzyme, we can go for the plastic degrading bacteria which will synthesize different type of enzymes which can degrade the plastic, or you can go for the bioplastic also. Then bio-based biodegradable materials, number of biodegradable materials we can construct which are bio-based, which are obtained from the nature, and in this way they will be degraded naturally. कंस्ट्रक्टेड वेटलैंड्स विथ माइक्रोबियल फिल्ट्रेशन. अब ये वेटलैंड्स बहुत इंपॉर्टेंट है. वेटलैंड्स जो है, वेटलैंड्स इट इज द मिक्सचर ऑफ सॉइल, माइक्रो ऑर्गेनिजम्स एंड प्लांट. और ये जो वेटलैंड्स होते हैं, दिस वेटलैंड्स आर यूज्ड एज अ कार्बन सिक्वेस्ट्रेशन एजेंट. Carbon sequestering, they will utilize more carbon from the environment and release less कार्बन डाइऑक्साइड इन द नेचर. सो ऑटोमेटिकली जो ग्रीन हाउस गैसेस है इनको कंट्रोल हम लोग वेटलैंड कंस्ट्रक्टेड वेटलैंड से कर सकते हैं. तो डिफरेंट टाइप ऑफ वेटलैंड हम लोग इंजीनियर कर सकते हैं विद द हेल्प ऑफ माइक्रो ऑर्गेनिजम्स, प्लांट्स एंड सॉइल, एंड इन दिस वे वी कैन ओवरकम द एनवायरमेंटल इश्यूज. देन कार्बन फिक्सिंग माइक्रोबियल सिस्टम्स, नंबर ऑफ माइक्रो ऑर्गेनिजम्स जो होते हैं जो कार्बन फिक्सिंग करते हैं, ऐसे माइक्रो ऑर्गेनिजम्स का यूज हम लोग माइक्रोबियल सिस्टम्स में कर सकते हैं और इसकी मदद से हम लोग एनवायरमेंटल प्रॉब्लम्स को सॉल्व कर सकते हैं. नाउ गोइंग टुवर्ड्स द क्लाइमेट चेंज मिटिगेशन. अभी हम लोगों को पता है कि क्लाइमेट बहुत जल्दी जल्दी चेंज हो रहा है, कोई भी मौसम में कुछ भी चल रहा है. लाइक समर है तो अभी हम अप्रैल की शुरुआत में भी नहीं पहुंचे, मार्च में हम लोग, और आज हम लोग देख रहे हैं तो टेम्परेचर 45 डिग्री तक चल रहा है. तो अभी ये जो क्लाइमेट चेंज हो रहा है, क्लाइमेट चेंज हो रहा है एज अ रिजल्ट ऑफ CO2 एमिशंस, डिफरेंट टाइप ऑफ ग्रीन हाउस गैसेस एमिशंस की वजह से हो रहा है. अब ये जो क्लाइमेट चेंज इम्बैलेंस है ये क्यों हो रहा है? तो राइजिंग एटमॉस्फेरिक CO2 लेवल, इंक्रीज मीथेन एमिशंस, डिफॉरेस्टेशन एंड सॉइल कार्बन लॉस, इंडस्ट्रियल पोल्यूशन, एक्सट्रीम वेदर इवेंट्स, बहुत सारे वेदर इवेंट्स जो हो रहे हैं वो एक्सट्रीम हो रहे हैं, उनकी वजह से क्लाइमेट डिसबैलेंस हो रहा है. So, as a result of this, disruption of natural microbial carbon and methane cycles, जो natural microbial carbon and methane cycle रन हो रही है एटमॉस्फेयर में, ये सभी डिस्टर्ब हो चुकी है. नाउ अगर हम लोग जाते हैं कि क्लाइमेट स्टेबल सिस्टम कैसे बनाना है, तो इसके लिए हमारे पास में बहुत सारे रास्ते हैं, लाइक मिक्स्ड क्रॉपिंग एंड एग्रो फॉरेस्ट्री. हम लोग मिक्स्ड क्रॉपिंग कर सकते हैं और एग्रो फॉरेस्ट्री को बूस्टअप कर सकते हैं, सो दैट सॉइल कार्बन स्टोरेज विल बी मोर. The soil will store more carbon inside it, and the CO2 which is emitted in the atmosphere will be automatically converted to the soil carbon storage. Then we can go for the organic farming, in which the soil microbial diversity will be enhanced. Then we can go for the different type of sacred forest which is acting as a natural carbon sink. अभी ये जो sacred forest होते हैं, sacred forest क्या करते हैं, they will consume more CO2 from the atmosphere and release very less CO2 in the atmosphere. So in this way they are acting as a carbon sink. Then we can go for the biomass recycling, control carbon cycling. अगर हम लोग बायोमास रिसाइकलिंग करेंगे तो नेचुरली क्या होगा, कार्बन साइक्लिंग जो है ये कंट्रोल में रहेगी. एंड नेचुरल वेटलैंड्स, वी कैन इंजीनियर नेचुरल वेटलैंड्स. नाउ ट्रेडिशनल इंस्पायर मॉडर्न माइक्रोबियल क्लाइमेट सोल्यूशन हम क्या करेंगे, तो इसमें हम लोग सबसे पहले ये कर सकते हैं, वी कैन प्रोड्यूस बायोचार्स. बायोचार्स आर द activated charcoals which are obtained from different type of biological products. तो ये जो biochars होते हैं, this biochars can be integrated with microbial inoculants and it will enhance the soil microbiome for carbon sequestration. Then we can go for the methane oxidizing bacteria for emission reductions. methane oxidizing bacteria का use कर सकते हैं हम लोग emission reduce करने के लिए. algal carbon capture systems are also there. We can implant the algal carbon capture system so that ये जो algae है, this algae will utilize more CO2 from the atmosphere and it will give oxygen to the atmosphere. तो algae जो है, algae can act as a carbon capture system. Then regenerative agriculture with microbial consortia. microbial consortia का यूज करके हम लोग regenerative agriculture कर सकते हैं. तो इस तरीके से अगर हम लोगों ने ये सब चीजें की, तो माइक्रोबियल जो आज के तारीख में जो क्लाइमेट चेंज हो रहा है, ये क्लाइमेट चेंज के लिए हम उपयोगी सॉल्यूशन ढूंढ सकते हैं विद द हेल्प ऑफ ट्रेडिशनली इंस्पायर मॉडर्न माइक्रोबियल सिस्टम. नाउ कमिंग टू द एआई, जो आज हम लोग बात कर रहे हैं एआई. अभी एआई आर्टिफिशियल इंटेलिजेंस. नाउ हाउ वी कैन यूज दिस आर्टिफिशियल इंटेलिजेंस in overcoming different type of problems. So माइक्रोबायोलॉजी ओरिएंटेड एआई मॉडल्स अगर हम लोग देखते हैं, तो नॉलेज एंड सिस्टम इम्बैलेंस. आज हम लोगों को पता है कि नॉलेज और सिस्टम, इनके बीच में इम्बैलेंस है. जो नॉलेज हमारे पास में है वो सिस्टम के पास नहीं है, या सिस्टम के पास है वो हमारे पास नहीं है. ये इम्बैलेंस जो है, इसको हम लोग एआई की मदद से सॉल्व कर सकते हैं. कॉम्प्लेक्स एनवायरमेंटल डेटा नॉट फुल्ली अंडरस्टूड. बहुत सारा एनवायरमेंटल डेटा हमारे पास है, लेकिन इसको फुल्ली अंडरस्टूड करने के लिए हम नहीं सक्षम है. क्लाइमेट एंड माइक्रोबियल इंटरैक्शंस डिफिकल्ट टू प्रेडिक्ट. क्लाइमेट हम प्रेडिक्ट नहीं कर सकते, माइक्रोबियल इंटरैक्शंस क्या होंगी ये हम प्रेडिक्ट नहीं कर सकते. एग्रीकल्चरल डिसीज आउटब्रेक्स, नंबर ऑफ डिसीजेस आर देयर विच आर रिलेटेड विद द एग्रीकल्चर. अब ये जो डिसीजेस है, इसका आउटब्रेक कब होगा, कैसे होगा, ये हम नहीं बता सकते. गट माइक्रोबायोम डिसऑर्डर्स इंक्रीजिंग डे टू डे. हमारा जो गट माइक्रोबायोम डिसऑर्डर्स है, ये इंक्रीज हो रहा है. एनर्जी एंड वेस्ट सिस्टम लैकिंग ऑप्टिमाइजेशन. ये भी बहुत ऑप्टिमाइजेशन जो है एनर्जी एंड वेस्ट सिस्टम, इसका ऑप्टिमाइजेशन लैकिंग है. तो ये सब जो प्रॉब्लम्स है, ये प्रॉब्लम्स को हम एआई की मदद से कैसे सॉल्व करेंगे. सो इन केस ऑफ एआई, ट्रेडिशनल सिस्टम यूज ऑब्जर्वेशन बेस्ड फार्मिंग. पुराने जमाने में हमारा जो फार्मिंग होता था, ये ऑब्जर्वेशन बेस्ड होता था, कि किसान जो है वो ऑब्जर्व करता था कि आज क्लाइमेट क्या होगा, उसके हिसाब से भाई 15 दिन में क्या क्लाइमेट होगा, फिर उसको खेत में बुआई करना है कि नहीं करना है. सो ट्रेडिशनल सिस्टम में ऑब्जर्वेशन बेस्ड फार्मिंग होता था. सीजनल प्रेडिक्शन मॉडल्स आर देयर इन केस ऑफ पास्ट टाइम. कम्युनिटी हेल्थ प्रैक्टिसेस जो है, ये भी हम लोग करते थे. नेचुरल फर्मेंटेशन मॉनिटरिंग हम लोगों को नेचुरली करना पड़ता था. इकोलॉजिकल बैलेंस ट्रैकिंग, ये भी हम लोगों को मैन्युअली करना पड़ता था. अब क्योंकि अब हम लोग एआई के युग में है, तो ये सारी चीजें जो है, इसको हम लोग इंटीग्रेट कर सकते हैं एआई से. सो एआई माइक्रोबायोलॉजी इंटीग्रेशन. अब हम लोग क्या करेंगे एआई की मदद से, एआई असिस्टेड सॉइल माइक्रोबायोम एनालिसिस। एआई असिस्टेड, तो एआई जो है, एआई की मदद से हम लोग बहुत सारे अल्गोरिदम्स बना सकते हैं, और ये अल्गोरिदम्स जो होते हैं ये सॉइल माइक्रोबायोम एनालिसिस करते हैं कि सॉइल जो है, सॉइल के अंदर कौन से माइक्रो ऑर्गेनिज्म ज्यादा है, कौन से कम है, उस सॉइल का टेक्सचर बढ़ाने के लिए उसमें कौन से माइक्रोबायोम हमने इम्पार्ट करना चाहिए, उस सॉइल की फर्टिलिटी बढ़ाने के लिए हमने उसमें और कौन से पोषक तत्व देने चाहिए। ये सारी जो चीजें हैं इसको हम लोग एआई के साथ में एआई असिस्टेड एनालिसिस कर सकते हैं। अभी ये जो एआई है इट इज हैविंग अ ह्यूज बेनिफिट इन एग्रीकल्चर, लाइक एआई ड्राइवन ड्रोन्स, सैटेलाइट्स एंड सेंसर्स आर देयर। अब ये जो एआई ड्राइवन ड्रोन्स, सैटेलाइट्स और सेंसर्स होते हैं, इसकी मदद से हम लोगों को 24/7 क्रॉप मॉनिटरिंग होती है, क्रॉप को मॉनिटर कर सकते हैं। इट विल डिटेक्ट द पेस्ट एज अर्ली एज पॉसिबल, वीड्स जो हम लोगों को ह्यूमन आई को नहीं दिखाई देते, ये एआई पावर्ड कंप्यूटर बेस्ड सिस्टम से हम लोगों को पेस्ट, वीड्स ये बहुत जल्दी पता चल जाते हैं कि हमारे खेत में कौन सा पेस्ट जो है वो फैलने वाला है। देन वी कैन ऑल्सो गो फॉर मशीन लर्निंग फॉर डिसीज आउटब्रेक प्रेडिक्शन। अभी डिसीज आउटब्रेक प्रेडिक्शन के लिए मशीन लर्निंग इज देयर। अब ये जो मशीन लर्निंग होती है इट इज हैविंग द analysis of diverse data sets like climate patterns, it will analyze the climate patterns, human mobility and social media trends। ये सभी का यूज मशीन लर्निंग में किया जाता है, और ये मशीन लर्निंग जो होती है ये हम लोगों को प्रेडिक्ट करके देती है कि कौन सा डिसीज का आउटब्रेक हो सकता है। तो डिसीज आउटब्रेक प्रेडिक्शन ये हम लोग मशीन लर्निंग की मदद से कर सकते हैं। उसके बाद में हम लोग क्लाइमेट मॉडलिंग कर सकते हैं। क्लाइमेट मॉडलिंग जो होता है इसमें हम लोग क्या करते हैं, एआई इंटीग्रेट क्लाइमेट साइमुलेशंस विथ एपिडेमियोलॉजिकल डेटा टू फोरकास्ट आउटब्रेक। आउटब्रेक को फोरकास्टिंग करने के लिए क्लाइमेट साइमुलेशंस बनाए जाते हैं और उसको इंपार्ट किया जाता है एपिडेमियोलॉजिकल डेटा के साथ। तो एपिडेमियोलॉजिकल डेटा प्लस क्लाइमेट साइमुलेशंस, इनका इंटीग्रेशन करके हम लोगों को क्लाइमेट मॉडलिंग करते आती है। और ये क्लाइमेट मॉडलिंग जो होती है, जस्ट अ मिनट, क्लाइमेट मॉडलिंग जो होती है, विद द हेल्प ऑफ दिस क्लाइमेट मॉडलिंग वी कैन go for different type of predictions which are related with the disease, which is related with the agriculture. Then AI guided probiotic personalization is also there। जैसे मैंने बताया कि पर्सन टू पर्सन गट माइक्रोबायोम जो होता है डिफर करता है, हर एक पर्सन के अंदर जो गट माइक्रोबायोम रहेगा वो डिफरेंट रहेगा। तो एआई गाइडेड प्रोबायोटिक पर्सनलाइजेशन पे हम काम कर सकते हैं। एआई जो है इसकी मदद से हम लोग पर्सनलाइज मेडिसिन बना सकते हैं जो कि उस पर्टिकुलर पर्सन के लिए अप्लीकेबल रहेगा, उसकी डाइट कैसी रहेगी, उसने कौन से प्रोबायोटिक यूटिलाइज करना चाहिए, उसका जो डे टू डे सिस्टम है वो कैसा रहेगा। उसके बाद में हम लोग जा सकते हैं स्मार्ट बायो रिएक्टर्स विच इज ऑप्टिमाइज यूजिंग प्रेडिक्टिव अल्गोरिदम्स। बहुत सारे प्रेडिक्टिव अल्गोरिदम बना के हम लोग स्मार्ट बायो रिएक्टर्स बना सकते हैं। अभी ये जो स्मार्ट बायो रिएक्टर्स होते हैं दे आर हैविंग द sensors and predictive machine learning system। So with the help of that the self adaptive system is formed। और ये जो self adaptive system होती है इसमें हमको मॉनिटरिंग करने की जरूरत नहीं है, ये ऑटोमेटिकली मॉनिटर करती है कि बायो प्रोडक्ट कब बन गए। और ये बायो रिएक्टर्स जो होते हैं दे आर हेल्पफुल इन सॉल्विंग डिफरेंट टाइप ऑफ प्रॉब्लम्स। सो इन दिस वे एआई जो है, एआई की मदद से हम माइक्रोबायोलॉजी में बहुत सारा वर्क कर सकते हैं integrating with the microorganisms। So in case of agriculture, in case of health, in case of probiotics, in case of environment sustainability, in case of industrial production, we can go for the help of AI and we can impart different type of algorithms, we can go for different type of personalized medicines, and with the help of this AI we can सॉल्व द डिफरेंट टाइप ऑफ प्रॉब्लम्स। बट ये जितना भी हम लोग एआई में वर्क करेंगे इट इज रिलेटेड विद द ट्रेडिशनल नॉलेज, क्योंकि ट्रेडिशनल नॉलेज जब तक हम इसमें यूज नहीं करेंगे तब तक हम एआई की मदद नहीं ले सकते। सो इन दिस वे द ट्रेडिशनल नॉलेज इज वेरी हेल्पफुल टू सॉल्व द मॉडर्न डे प्रॉब्लम। एंड with this slide I end my presentation here। So thank you very much for the silent listening, and if you are having queries it is open for all।
Yes, participants have any queries regarding on a present topic, a traditional insight for modern sustainability. Madam, one question from my side.
Yes, sir.
Whenever in a present scenario, everyone looking for the AI,
Yes.
and really this AI will be the future outbreaks of the bacterial or the fungal infections, and we are
Sir,
trust on that.
सर प्रेजेंट डे वी कांट से कि एआई जो है फ्यूचर में हम लोगों को बहुत मददगार रहेगा, लेकिन हाँ ये बता सकते हैं कि एआई जो है, एआई हम लोगों को एक बेहतरीन सॉल्यूशन दे सकता है अगर हम लोग डिफरेंट टाइप जो हमारे पास में नॉलेज है उस नॉलेज को हम एआई में यूज करते हैं, तो ये एआई की मदद से हम लोगों को प्रेडिक्शंस जरूर मिल सकते हैं सर।
लेकिन
क्योंकि
मैडम आज
ह्यूमन,
प्रेजेंटली,
यस सर,
यस कंटिन्यू मैम कंटिन्यू।
यस, क्योंकि क्या है कि एआई में बहुत सारे अल्गोरिदम्स हम लोग यूज कर सकते हैं प्रेडिक्शंस के लिए। अब जैसे एक एग्जांपल अगर हम लेते हैं कि एक फील्ड है, वहां पे फार्मर जो है, फार्मर को उस उसके फील्ड में कौन सा पेस्ट फैलेगा ये कब पता चलेगा? जब उसके झाड़ खराब होने शुरू होंगे, उसको कोई ना कोई झाड़ में कुछ डिसीज दिखेंगे, येलोइंग दिखेगा लीव्स का, उसके बाद। लेकिन अगर हमने एआई की मदद ली और एआई में ये सारी चीजें पहले से हमने मशीन लर्निंग में फीड करके रखी, तो एआई हम लोगों को प्रेडिक्ट करके दे सकता है कि आपका जो प्लांट है इसमें ये चेंजेस होने शुरू हो चुके हैं। तो एज कंपेयर टू ह्यूमन, एआई की मदद से हम लोगों को ये सब चीजें फास्ट मालूम पड़ सकती है। तो 100% हम उस पर रिलाई नहीं कर सकते कि फ्यूचर पूरा एआई पे बेस्ड होगा, लेकिन हाँ, एआई इज वन ऑफ द गुड ऑप्शन फॉर द प्रेजेंट डे सिचुएशंस, ये बोल सकते हैं।
ये हो सकता है, इट बी द बेटर टूल, but we can't
Yes,
be reliable. We can't be
yes,
reliable.
yes. We
Yeah,
can't rely
just now the
solely on AI. हाँ,
yeah, yeah. Justly
100%
we
हम
have
उसके
the
ऊपर
n number
रिलाई
of
नहीं कर
satellites.
सकते। Yes,
Just India having the n number of the satellite regarding with the agriculture, regarding with your environment. So whenever there are the certain messages coming from the meteorological department on the same day,
yes,
there should not
yes,
be any rain. Can it is happening
yes,
in future also with the AI?
yes, sir.
Thank you, madam, for your wonderful lectures. And at the same time, today, the 8th March, Women's Day. So I wish all the women on this great day, Women's Day, a happy Women's Day to all joined here on this platform as well as on the YouTube streaming also. I really thankful to Dr. Pachori madam for his wonderful lectures on the traditional. His presentation is very nice in all the aspects. He takes the each and every the core sectors, right from the health to the environment. On the behalf of a national conference, organizing secretary, I really thanks for your wonderful lecture. At the same time, you are spending your valuable time with us. Thank you, madam. Thank you.
Thank you very much, sir. I also thanks the organizer of this conference to give me a chance to speak in front of all the audience. So thank you very much.
Yeah, thank you, madam. Now over to Dr. Ashok Davande, sir. Davande, sir, are you
Hello.
there?
Yes, sir. Yes,
Yeah.
I am here.
Okay. So the third technical session, I requested Dr. Davande, sir, to introduce the third keynote speaker on the second day national conference. As a convener, as well as a head department of microbiology, Shankarrao Bhair Patil Mahavidyalaya Pulgaon, IQAC, Dr. Ashok Davande, sir, please introduce today's third keynote speaker.
Thank you, sir. First of all, I welcome today's resource person, Dr. Bipransh Kumar Tiwari sir. It's my great pleasure to introduce Bipransh Kumar Tiwari sir, who is currently serving as Assistant Professor and Head Department of Microbiology at North Bengal St. Xavier College, Jalpaiguri, West Bengal. He also serves as IQC coordinator of the institution and visiting faculty in the Department of Biotechnology at University of North Bengal. Dr. Tiwari has made remarkable contribution to the field of microbiology. Earlier, he served as the state president for West Bengal of the Microbial Society India. He is recipient of several prestigious recognition, including Senior Teacher Award at national level from Microbial Society India and Young Scientist Award presented at second international conference held at Sanjeevani Arts, Commerce and Science College, Kopargaon. Dr. Tiwari has actively engaged in research and is currently supervising PhD scholar. His research interests include medical microbiology, phytomedicine, computer-aided drug design, antimicrobial development, and bacterial column sensing. He has impressive academic record with 25 research publication in a reputed journal, has edited 5 books, and contributed 10 books chapter to academic publication. Additionally, he has delivered more than 20 lectures as a research person in national and international seminar and workshop. Dr. Tiwari is a lifetime member of Association of Microbiologists in India, the Biotechnology Society of Nepal, and Micro Society India. He also served as reviewer for several high-impact scientific journals. With this brief introduction, it's my privilege to invite Dr. Bipransh Kumar Tiwari sir to share his invaluable insight with us. Thank you, sir.
So my slide is visible now, sir?
Yes, sir, it is visible.
So thank you, Dr. Ashok sir, for the warm introduction. It is truly my privilege to be part of this esteemed academic event. I express my sincere gratitude to the organizing committee for extending this invitation. And my special thanks to Dr. Indus sir, MBSA President, and Dr. Rakesh sir for the kind gesture and support. We all know that we are living in an era of the amazing scientific progress, but also with some major health crisis. And this crisis makes us very clear that the future of healthcare will depend on how smartly we understand the microbial world and how creatively we use that knowledge. So instead of relying on the traditional antibiotics, we need to look at the new and the innovative, especially the pathways by which the microbes use. So these pathways may help us to develop the better, safer, and we can say the more sustainable solutions for the future. So therefore, today I am going to discuss antimicrobial resistance crisis. Can antivirulence therapy and artificial intelligence offer a sustainable solution? So I'm mainly working on the screening that is planned for the anti-colon activity and also documenting the traditional medicinal knowledge of the medicinal plants and natural, you can say, the compound that had the ability to kill or to inhibit the bacterial infections. Am I audible to all of you? Hello?
Yes, sir, audible.
Thank you so much. So let's begin with what the greatest challenge and the challenge of global threat. So let's begin with the and antibiotics have been one of the, we can say, the greatest discovery in the medical history. We can see that the first antibiotic, that penicillin, given to the patient in 1941, and there were the introduction of the new antibiotics. Okay, and this introduction of the antibiotics, why the greatest discovery in the medical field? Because it saved millions of lives. And fortunately, we have the antibiotics against all the infections. But when you see the right side, unfortunately, of the antibiotics are now, we can see, you can see some you will see the pattern here. We can observe here that in 2000, most of the antibiotics were effective, means they were able to kill the bacteria for those they were designed. But now the scenario has totally changed in the 2025 or 26. The scenario is just reverse. Okay, you can see that now most of the antibiotics are not effective, but you can see most of them, means bacteria are growing stronger, and now it's becoming harder to treat. So now let me define antibiotic resistance. So we define that now the bacteria develop the ability to defeat the drugs designed to kill them. Most of the bacteria resistance published in the WHO that support 9 million deaths annually due to this AMR. And the Lancet study found that 1.27 million people died only due to this antibiotic resistant bacterial infection. So these two reports, I think, that can justify that how this one is the global threat. So if we do not take action now, then obviously it will be the threat in the future. So you can see one data produced by the WHO. Different cholera, measles, diabetes, cancers. I used the cancer 8.2, but you can see the scenario that death from the antimicrobial resistance are forecasted for the 2050 10 millions. Okay, so now we can understand that how this one is the biggest crisis or biggest challenge in front of the medical field now. But when you hear the term the antibiotic resistance, so this is not the new term, not the new phenomenon. Well, the report published, they have reported that the 30,000-year-old bacteria had been recovered that bacteria developed in an arms race that is known for the billions of years. So they found the genes for the beta-lactamase, they found the genes for the tetracycline resistance, okay, they found the genes even for the vancomycin. So these genes are already present in the bacteria, not the new phenomenon. But our huge novel of antibiotics is amplifying the phenomenon dramatically. So the application of the suppose this one is the population of bacteria. So here we can see this population. So most are the sensitive ones, only the few bacteria are the resistance. But when we expose this population with antibiotic, then what will happen? All the sensitive will be killed. Okay, which one can resist resistance now the few generation and this option will be full of the one. So we have selected the resistance only sensitive one. And by exploring the bacteria with antibiotics, we are creating the selective pressure. And due to the selective pressure, there is the emergence of the antibiotic resistant strains. So now just go to this cartoon. Here we can see that I have a flu and I shall visit a doctor. So many people, means when often we ask to our relatives or to our friends, I have a flu, I shall visit a doctor. Often we get this suggestion that no need to visit a doctor. That is very normal. But when I ask a responsible doctor that I have a flu, you should get some rest. It means by this scenario, you overuse of antibiotics is amplifying the antibiotic resistance. Now the question is how to minimize the load of antibiotic for treatment, because still today the antibiotics are critically important to control the bacterial infection or to treat the disease. Still it is critically important. So we cannot ignore that. So now we have to figure out how to minimize the load of antibiotics. And second, that do we have to think of some alternatives? So with these two questions, I will continue my discussion. So just I want to show one more cartoon here to make you understand that what are the alternative approaches and how can we think about the innovative approaches to control or to combat or to control the emergence of antibiotic resistance. So here we can see a person with the and this may be one example of the virulent bacteria, means this represents a harmful bacteria. So when this harmful person will be in front of you, then we have the two choices only. To protect ourselves, we have to kill this person. And we can see in this first that the killing of this person. So now this person cannot harm you or harm us. So now if you compare this with the antibiotic, most of the existing antibiotics, what the objective of antibiotics does to eliminate or to eradicate the bacterial population. So here we can see the mechanism of antibiotic actions are inhibition of the protein synthesis. It's the pathways or the which are required for the survival of the bacteria. So inhibition of the nucleic acid synthesis, inhibition of the cell wall synthesis, disruption of the membrane, and the blocking of pathways of the inhibitor mechanisms. So all these are required for the survival of bacteria. So now the bacteria try to resist the antibiotic or try to evolve themselves to adopt or to survive the presence of the antibiotic. The second choice that we have, just snatch out or take away this weapon from some person. Still the person is alive, but it cannot harm us. So this person is now harmless. We are not afraid of this person, we are afraid of the weapon, person with the weapon. So now if you somehow take away this weapon from this person, this one will become harmless. Okay, so now this person cannot harm, so now this will harm. Somehow, if you don't kill the bacteria, just inhibit the virulence factor or just inhibit the ability of bacteria pathogenesis. Okay, now there is no selective pressure, we can say, and bacteria will not evolve themselves to resist or to survive in presence of the antibiotics. So this approach is known as the anti-virulent therapeutic strategy. That interferes the virulence without killing the bacteria. Okay. So this cartoon I have shown just to make you understand that part of the difference between the killing bacteria and simply disarming them. So now the next question, we can see that antibiotics, the traditional method that we apply to treat the bacterial infection, and the new approach that we can look into, that the antivirulence drugs. So this and in the antibiotics, it kills bacteria, it targets the bacterial group, so it creates the high selective pressure for survival, and with this one there are the high chances of the resistance to antibiotics. But if you have for this antivirulent drug or antivirulent approach, we can see inhibition of the virulence, not the growth of the viability. low selective pressure for the survival, low resistance and tolerance to antibiotic killing, clearance of pathogen. I will discuss that still there is means issue with the clearance of pathogen by going to the antiviral therapy and the preservation of the beneficial flora. And the chances of the high resistance, they do not like the chances of the emergence of the resistance against the antiviral drugs. So now we understand that how the from 2016 to the 2025. So what the principle of the antivirulence? So not to kill the bacteria, just to disarm the bacteria or to inhibit the virulent property of the bacteria. So now the question, how to inhibit the virulent property or the virulent character of the bacteria. So normally next person to communicate. We all know that yes, the bacteria communicate. So bacteria each other and the language is the chemical. Now you can see just like the components we know at the social. We know that the four components of the communication. The first is the dialect, the language that we use to communicate. Second, we hear, we can recognize, and then we respond. And then after the recognition, it is after responding, they have to take action, what to do the next. And then the social behavior. Similar in the bacteria, there is the communication. Dialect, they use the chemical compounds. They use the binding of chemicals to the receptor protein, the recognition. They take action by the expression of genes. And this social behavior is known as the sensing. So now see at the dialect, so we can see that most of the gram-negative bacteria use chemical compounds known as the autoinducer. These are the small diffusible molecules. So most of the gram-negative bacteria use the autoinducer, acyl homoserolactin as the dialect. And the second, we can see that here, the gram-positive bacteria, they use the autoinducer peptides. And just some understand among us, some understand the Hindi, some understand the local language, but English is the common one. So this AI2, so this one is common, gram-negative and gram-negative, AI2. So now we can see that this is the population density dependent. So autoinducers expressed from the bacteria, you can say that it can diffuse out from, means it can cross the cell membrane. At the low cell density, you can see there is no tower, means there is no networking, means there is no any communication between the two bacteria. But when the population increases, and with the increase in the population, there will be the increase in the autoinducer. So when this autoinducer reach to the threshold value, at the threshold value, then they diffuse into the receptors, and now the bacteria starts to come. So the bacteria are very clear. So now we can see that the bacteria the virulence, bisense, swarming, pigments, biofilm formations, all these are the group behaviors and density dependent. So now we are able to define the term the quorum sensing. So this quorum sensing is a cell-to-cell communication mechanism regulated by the small diffusible signaling molecule termed autoinducers, which allow the bacteria to respond and adjust their needs in a population density dependent manner by altering the expression of the molecules. So this control and production, some cases the antibiotic production by formation of the surface molecule. So now the see that how it's controlled. So I'm just going to explain the most studied system, that Lux-I system found in the gram-negative bacteria. So there the Lux-I and this Lux-I is the acyl homoserine lactose synthase. So it produces from this Lux-I the synthesis and diffusion. This Lux-I the receptor. So when this is through the expression of all the genes which are associated with the quorum sensing. Okay. So the expression takes place only the auto-inducer reach to the threshold concentration, means the bacteria reach to a quorum population. So these are all the multiple gene expression causes the virulence antibiotic biosynthesis, but today we will mainly focus on that the virulent property of the biofilm formation. So now we understood that this quorum sensing control the timing, we can say, and the production of the virulence factor. So now how to control the we have to stop the bacterial communication. The next question, how to control or stop bacterial communication? So we have to disrupt the communication that exists among the bacteria. So breaking the code of the bacterial language may one day yield an alternative or the traditional antibiotics. So three strategies that we apply here: targeting the signal generation, with the signal precursor of visual producer, we can inhibit that one. Targeting the agent signal dissemination, and the third is the targeting the signal receptor. So these three strategies mainly apply for stopping the communication in bacteria, because we know that this communication is responsible for the virulent property. So by controlling what we are, instead of killing them, we reduce the selective pressure. Instead of killing them, we avoid the pushing bacteria to develop the resistance, and we make infections means more easy for the immune system. So you can see that the different material that we use, we like that other compounds, and the single compounds, the receptor or of the dissemination of the signal. So as I already mentioned that I mainly work on the screening of the phytocompounds on the plant extract for the quorum sensing. So due to the large diversity and the biochemical complexity, natural products act in the different ways against the quorum sensing. So first we have targeted the guava leaves. This is the very common plant. We can see that. And we use the model organism Malaysia, and the both are the gram negative bacteria. So this the biofilm formation, or swarming motility, swimming motility, all these are associated with the quorum sensing phenomenon in Pseudomonas. So these two model organisms we use for our. So here we can see in presence of the guava leaf, there is the growth of the bacteria. This is without the control, means no guava leaves is present. So you can see that Chromobacterium violacea produced the violation that is in color, but in presence of the guava leaf, you can see there is growth of the huge growth of the Bacterium violacea, but there is no production of color, the violacea. Here you can see in the that there is the clear, not means you can say the transparent. This is the opaque, but there is no color. So this guava leaf extract acts as the potential inhibitor for what? The quorum sensing regulated phenotypes in the Chromobacterium violacea, that is the violation products. Now we have seen that we perform the experiment to that the concentration, we can see that with increasing concentration there is the decrease in the violation products. Okay. And after the 400 there was the saturation. Okay. And then we for the growth curve, so what we found that in control experiment, there was no any difference in the growth curve of the in presence or in absence of the guava, means there was no effect of the guava leaf extract on the control on the growth of bacteria. But simultaneously we evaluated or we determined the percentage of violation. We can see this is for the control and experimental. There is no production of the violation. So at the 400 microgram per ml, 85% inhibition in violation production in presence of the guava leaf extract. But there is no any effect on the growth of bacteria. Now we can see the inhibition of the swarming motility. You can see this one is the in presence of the guava leaf. There was no any swarming motility in the Pseudomonas. So this swarming motility also associated with the quorum sensing in gram means Pseudomonas aerogenes. Then we went for that the transcriptomic analysis that what are the genes affected by the quorum sensing. So you can see that the genes expressed exclusively in constants of guava or the 1 to 9. Gene expressed exclusively in experiment it was 32, and gene expressed in both samples 3025. Expressed in both sample it was the 32, not expressed in the both sample 243, because we know that the 20% of the genome means the genes associated with the quorum sensing, and 5 to 10% of the protein that associated, 2 to 5% of the genomes, and the 20% of the protein associated with the quorum sensing. So here we can see that this control says 3025. So this in control system only expression means 20% of the genes are down regulated in presence of the guava leaf extract. So you can see in both sample 3025, in control only 1229 gene expressed exclusively, means these are not present in the experimental one. So what we found that 19 to 20% of the genes were down regulated in presence of the guava leaf extract. Here we can see that up to only 6, but 196 down regulated in presence of the guava leaf extract. Then we classified that the genes in which class it falls, and then to test that, confirm that this quorum sensing inhibitor attenuate the virus relation. So Chromobacterium violacem induced cytotoxicity in HepG2 cells in vitro will be assessed. So we have infection of the HepG2 cells assessed by the phase contrast microscope and the LDH assay. So what we found, we can see that in presence of this one is lysis of HepG2 cells after the 4 hour of infection of the Chromobacterium violacem, and this the inhibition of lysis in presence of the 400 microgram only GLE, and this one represent that in presence of the means when you treated the Chromobacterium violacem with the GLE, there was no inhibition of the cell. So here we can also see the LDH-ASH. Okay, so here only water, so there was no leakage. Here we can see only 400 microgram of GLEB. See only Chromobacterium violacem. So it infected the cell, and due to the lysis, and we can see the LDH, total LDH activity. But when we previously treated the Chromobacterium violacem with guava leaf extract, and then we try to infect the cell, now we can see the LDH activity reduced. Okay, it means it inhibits the virulent activity of the Chromobacterium violacem. Means guava leaf extract did not inhibit the growth of bacteria, but it successfully inhibited the virulent property of Chromobacterium violacem. And then in our second work, we have screened the 61 plants from the Darjeeling Hills, and we found that the 17 plants were able to inhibit the Chromobacterium production, but only there was no any effect on the growth of the bacteria at the 5. And finally, we found that the 3 plants, Stabile rivularis, Fageria nubicola, and Ashbukia nepalensis, these 3 were able to inhibit the violation without affecting the growth of the bacteria. So these are the results. So we can see that inhibition of the swarming motility, inhibition of the violation production and the processing. Then we did the GCMS and these are the phytocompounds we found for the phytocompounds that were also hide the fusone. You can see the hexahydro-3-methylene. So all these are the found in the GCMS analysis. Okay, so among the 61 plants, 17 were able to inhibit violation production, but only the 3 of the 5 plants, Stilbe rivularis, Fageria nubicola, and Osbechia nepalensis, showing those dependent inhibition in violation production with no effect on the bacterial growth, were considered the most promising plant with antiquorum property. And then after that, we when the plant extracts So the result, then we try to go for the nanotization of those plant extracts. So on the melastoma, we have nanotized it. So you can see this is the nanoparticles, characterization of the nanoparticles, and then we found that the nanoparticles were more effective than the only the phytocompound. You can see the result. There is the growth of huge growth of bacteria, but there is no production of the color, means phyocyanin in Pseudomonas aeruginosa. Here the violation in case of the Tomobacterium violation in presence of the nanoparticle. So these are all silver nanoparticles. Then this identified activity, we can see that this one control the violation progression taking place there, and 1/4 and this is the 1/2 value. We can say that this nanoparticle were able to inhibit the formation of biofilm. So this was my first part of the presentation. So what the takeaway that what is of the supreme importance in war is to attack the enemy strategy. So by using the antivirus therapy, we are just trying to attack or to trying to target the enemy strategy, means our enemy bacteria, how to inhibit only their virulent property, not to kill them. And by this way we can minimize the selective pressure, we can avoid pushing the bacteria of the resistance. Now, second part, that how can we use the AI for the AMR management or AMR study? So first of all, I would like to mention that I'm not an expert in the AI, means in the development of the AI models or the mathematical simulation, but for the last 1 year, so 1.5 years, I am trying to use the generative, mainly the generative AI for the research, and I'm using those AI tools to enhance my data analysis. To some extent, I use to develop the models that I can work on the antibiotic resistance. But I'm going to serve only the theoretical part. So the paradigm shift, the transitioning from the slow observation laboratory science to rapid predictive data science only possible by the AI. So we will say that how AI why can we use the AI in the AMR management, AI in the antibiotic development, machine learning for the resistance forecasting, and AI predictions in the disease management. So first, let me define that artificial intelligence is very that the two words are there. Artificial means not man-made, and the intelligence means ability to think. So when we use the term the artificial intelligence, means we train the machines to think or to like the human. So the discipline that creates the computer systems capable of activating is normally associated with the cognitive effort. Okay. So a computer program that can perform tasks that typically require the human intelligence. It means basically the study of training your machines, or machine means here the computer, to mimic the human brain and its thinking capability is the artificial intelligence. So mainly the three types of the artificial intelligence we use. That first one is the narrow AI. So this specializes in one area and solve one problem. We can see in the traffic signal, the camera is there just to find out that which one is breaking the rule, to capture the image. So this is a single task. So this is your narrow intelligence. And then the general intelligence, that is the generative AI, generative artificial intelligence. So this one refers to the computer that is smart as human. It can generate the idea, it can generate the means ideation, it can generate the means report. Whatever the human can think, this generative AI can think or can perform that task. And then the artificial super intelligence. So here the intellect that is much smarter than the best human brains, means it can think beyond the human brain, is your artificial super intelligence. So mainly that way ChatGPT and all the chatbot, these are the generative AI. So we have three terms, the artificial intelligence. So here we can see that which enable the machines to make the human behavior simple. Machine learning, so this one is the subset of AI techniques, which use the statistical method to enable the machines to improve with experience. And then the deep learning. So subset of ML which makes the computation of multi-layer neural network feasible. So now we know that AI is becoming very popular in education and these days, because the most of many teachers and students and the researchers are using AI tools to make the learning easier and to make it more interactive. Students and researchers are using the AI tools to understand the critical concepts, summarize the research papers, and data is also in the research writing and data analysis. So now this is crucial that artificial intelligence has emerged as a powerful tool to support the researchers and healthcare professionals in understanding and managing the AMR. So first, you can see that applications of healthcare in monitoring the patients, in the guiding the surgical care, predicting the health, recommending the treatments, optimizing the operational process, and this can be justified by the number of the research articles published in the healthcare sector, especially in the surgery, radiology, cardiology, that within years there's a huge increase in the number of the publications on the AI on the different fields of the healthcare. So now the AI, the paradigm, the shift in the drug management. So one of the most important applications of AI is the prediction of antimicrobial resistance using ML model. So AI algorithm can analyze the large amount of the genomic data set of the bacteria and can identify the genetic mutations responsible for the resistance or not. Then the machine learning models, they can predict whether a pathogen will be resistant to a particular antibiotic before the laboratory testing is completed or finished. So this will help the clinician to choose the most effective treatment more quickly. Or we can say that the cosmetic treatments may be possible use of the AI tool. And the other application of AI in drug discovery. So traditional antibiotic discovery is the time consuming as well as very expensive. So AI can screen the millions of chemical compounds in a short period of short time, and also can evaluate the drug properties of those compounds, and by this way they may help in the development of the new antibiotic, because we know that since 1987 there is no development of any novel class of antibiotics. And also this AI will play roles in the key in surveillance by analyzing hospital data, by analyzing records and data. The systems can track what the emergence and the spread of the resistance pattern in the particular area. And this will help the policy makers and healthcare providers to make the policy to develop the better antibiotic stewardship program. So this from reactive treatment protocol to the proactive data-driven biological intervention is possible by the use of the AI in AFR. So now the drug discovery of the halicin is first powerful antibiotic discovered and using the AI and the AI platform is the GENTRL. So here the in silico medicine to generate new compounds with the targeted antibiotic. So this one is possible by this platform GENTRL. And also we use the AI means for the antimicrobial susceptibility testing. So here we can see that the problem the traditional culturing is slow and the labor is, but so this AI algorithm genome is just to predict the antimicrobial susceptibility directly from the critical sample in a fraction of time possible by the genome testing. So now the question human being, but someone empowered by AI undoubtedly. Okay, so we have to be empowered with the AI tools, and we have to use the AI to enhance our potential. So now I'm going to just briefly discuss the generative AI, and then I will discuss few use of AI in the AMR surveillance from peer review articles. So here we can see that we can use the generative AI, means which can generate the ideas, generate the report and all. So mainly we use the ChatGPT, that is the generative pre-trained transformer, GPT-4, until the GPT-5 so integrating the electronic health records, sliding doors, improving the and so here I'm going to show a few prompt example that can use for our literature search, or you can use to get the some basic information about the antibiotics or AMR. So I have already mentioned that the generative AI is subset of AI that focus on the creating new data instances. So mainly we use all the chatbot, whatever we use the ChatGPT, Gemini, Copilot. Okay. So first example I'm showing the antibiotics for the we can see the this one can get the ChatGPT. Now the second question, how can we use generative AI for the education? So one example of the prompt is given. Besides multiple choice questions, is the training to learn about the evidence-based strategies for antimicrobial stewardship implementation based the questions on the attached guideline. So we have attached this guideline, and now we can see that the example response is this one. So this way we can use the generative AI for the education purpose. The second prompt you can see based on the attached article, create a common clinical case scenario in which antibiotics are overused. Include the ASX past medical history of the presenting illness, any relevant information and information. So now we can understand how can you use AI enhance our unnecessary urine for automatic background. And there are several literatures about the most common side given here. The consensus is here, the scholar AI, the LCT is there, perplexity is there, connected papers are is there. Okay, so we can use all these AI literature search for the literature search. Now we can see the second prompt here. Provide the list of diagnostic stewardship interventions that aim to reduce the unnecessary urine culturing for asymptomatic bacteria. So like this way we can use the generative AI for the literature search for education purpose. Now, how the generative AI and then so we can see the dev guidance. Create the custom antibiotic stewardship GPT, develop new guidance documents using the prior example. For the education purpose, generate the quizzes from existing content, create the analogies to explain the concept, develop the infographics and presentation. Now the summarize short, long documents and articles, identify key messages from podcasts and videos, research, reform and literature, critique your scientific, translate to from English, manipulate the data. So all the generative AI we can use AI can generate lots of information or knowledge on the AMR. My objective just because most of the students and the research scholars are present here in this seminar, so they can use the generative AI for the development of means research, not the development for their education purpose. Now come to the machine learning. After the generative AI, the second stage is the machine. How can we train our machine? Okay. So the first here we can see the sample predicting that how to use the machine learning, how to train your machine for predicting. First information in the sample. It means you have to so at this lesson, we have to get the information together, the data of the clinical and the data, and also we have to collect it from the EHR, means electronic health records, and these we can include the informations of the previous antibiotic use, the prior resistance history, and the healthcare setting. And this data may help to our machine learning models to estimate the risk of resistance infections. And then the second is your culture positivity, where the collected sample is culture determined whether the microorganisms are present. Then third, we have to find a best for the identification. So that we can specific identification possible in a short period of time by the multi-top, that the right fingerprinting by using the PCR, which is, we can repeatedly determine the microbes species. And then we have to go for the antimicrobial susceptibility testing to means we have to perform to determine which antibiotic the pathogen is resistant or susceptible to. Okay. So this will help us to means clinicians or the researchers to make faster and more precise antimicrobial resistance or antibiotic treatment decisions. So now the machine learning process for the that we mainly use the right and then we assume that all the features are independent, means assuming feature independence and from each other. And this one is the simplest one. So simple, fast, accurate for the large data. So what we predict, we can predict the effective drug combination, estimating the probability of the effective treatments due to the AMR. So these means drug combination and estimate the probability this possible by using the naive Bayes means algorithm. And then the decision tree. So this algorithm helps us to take the decision, means not us machine take decision. So here describing what the features we in this system that we can classify. We can classify okay. So this important features will guide or its means machine to antibiotic selection. So here we can see that shorten the treatment means allocating the medical resources by estimating the AMR burden. And the most popular, the random forest. So decision tree using the random sampling to lower generalization errors. So basically what we trained, we improve the reliability by using the random forest, means we decrease the noise or we decrease the unrelevant information in the random forest. Means accurately predicting the multi-drug resistance tuberculosis. We can see support vector medicine by classification model that identifies the partitioning hypertension molecule. So predict the AMR phenotypes, other susceptible or resistance. So here you can see one example is antibiotic resistance in E. coli. So 90% means sometimes above means accuracy sometimes more than the 90%. And then the artificial neural, that is the network, it just the deep learning, we can say, not machine, it's deep learning. So architecture is by the human brain capable of complex pattern recognition. So in the deep learning, there is information layer by layer. So the information so it can think beyond the human brain. So the purpose screen how we can see the example is that how the predicting resistance by using the laboratory and the health report data. So the first is the suitability of data. So first you have to check the quality of the genomic data that we have gathered. Means before applying to the machine learning, you have to check the reliability of your data. So that is possible by genome assembly quality may be reliable. The data should have the balanced AMR phenotypes of node and population structures of, and then the encoding data. Encoding data means converted into the numerical or the mathematical format. It means 1% means 0. Genomic features that are detected the change the positioning single nucleotide okay, or the phenotypic susceptible resistance MIC. So this is the machine related matrix. Now the third is the feature selection. So this is the model independent selection with association test. So here among the thousands of the genomic features, many of which are not relevant, we can means exclude them one and we identify the most important genetic marker associated with the AMR. And then the ML algorithm selection. So we have discussed that we can go for the random for the SVM and the neural network. And then we have to train your machine and optimize. prediction accuracy. And this one application of AI to AMR surveillance, we can say that the first, the approach is the sequence presentation. So mainly in the sequence presentation, what we do, the gene-based encoding means the authors have checked that whether the AMR genes present or absent in the sample by aligning the sample to the established database. And then the mutation-based encoding means authors have compared it with the reference genomes and recorded whether there is specific mutation or not, and then compared it to the mathematical model that one has zero. The composition-based encoding and means the length and noting the presence set KML 1, 2, 3, 4, 5, so that 8 is absent, 7 is absent, 6 is present, and 6 more is present here. Then the second, this example that how the AI used for the ciprofloxacin MIC prediction. Okay, means they used, the authors have used, means this was the Pataki and colleague, they have used the AI model to predict the minimum inhibitory concentration, mean the minimum concentration of ciprofloxacin required to inhibit the growth of the E. Okay. And for training means they started with the massive data set and then they AI from the field, they have it can only the 4 most important predictors here we can say for future by using the random forest feature selections and all, and then they have predicted the AMIC value, means just after feeding only the 4 features into the second random forest, they predicted the MIC for the new E. coli isolates for unknown geographical region. But you can see that how the AI can help us. Experiment we have to perform, but for the data analysis, they have started with the 830K, means 83000 genomic features. And select the 4 features based from the MIC. The second is that for the so just isolated one area work. So there are the ARGs that you have to classify it, and then the 100 nucleotides you have to identify that whether they are present or not, and then normalize the bit scores to 4330 known ARGs. In this case only, I have taken from one paper, and then fed into the multi-class of the feed forward neural network. Here we can see, and then 30 PS for each AMR category. And here the AMR prediction from the HGT means the horizontal gene transfer. So here metadynamic is post gene removed, ARGs to find out that whether the ARG is presenting or not. And this one is the mobile genetic element. The mobile genetic element for and then study that filtered the raw reads and to remove the host. So mainly they performed this experiment on the chickens in across the China. Okay, and they found that here they trained the several machines. You can see the random forest. These are the training of the forest, and then they ultimately come to the predicted the resistance to the 10 antibiotics. Okay, so by this way, by using the different algorithm and the machine learning tools, the student is possible to mainly to manage AMR or the so here we can see the data scarcity. So the first challenge is data scarcity because we have you can see here the SMPH. Okay, but in many AMR studies, what the available data are incomplete. So this one is one challenge, that data scarcity and quality. The second, we can see the black box for the complex deep learning models suffer from low interpretability, hindering clinical transparency and trust in the decision making. Okay. The third is the generalizability. AI model training on demographic or the clinical significantly fail where apply to different global context. Means we have to develop the model, develop the ML. Okay. to the extent so that we can use the customized treatment for the patient. Now the infrastructure and cost. So it requires the advanced AI requires the massive computational power, capital, limiting access in resource poor setting, and obviously the most important is the ethics and privacy. So managing the informed consent, patient data privacy, legal liability. So when AI determines the treatment pathway, so all these are the you can see the challenging of using AI in the AMR and also in the healthcare center sectors. So the future of the multi, you can say the synthesis, AI alone cannot solve the AMR. It must be coupled with the rigorous experimental laboratory validation and the global health policy. Shifting from the reactive antibiotic deployment to a personalized predictive digital ecosystem tailored to patient's unique genetic profile and the pathogen's real-time evolutionary state. This is the of AI to provide the patient's unique genetic profile and the pathogen's real-time evolutionary state means customized medicines. So the take-home message from this future, artificial intelligence include many subfields, most of which are applicable to the antimicrobial stewardship, but it requires the high-quality data, approach to AI and stewardship, understanding both the benefits and the limitation. There are not only the benefits, there are also challenges and limitations. Generative AI holds promise, but technology is nascent. Okay, that's why I discussed the ChatGPT and all the chatbot. But generative AI, some extent we can use the AI for research. To keep up with the antimicrobial stewards and hospital epidemiologists can participate in training and model development on testing. So once a new technology roll over you, if you are not part of the steamroller, you are part of the road. So these are the few publications on the alternatives to antibiotics in the last 5 years. And I must acknowledge my collaborator, Dr. Manav Adhikari, and these are the two research scholars, Ms. Nitya Rai and Ms. Sangeeta Das, and the funding agency. I see them running the fund for running the project. So thank you for your attention. And once again, thank you for this kind invitation in this seminar. Hello.
Hello. Thank you, sir, for your
Thank you so
wonderful
much.
presentation.
Thank you so much, sir.
So your presentation and your ideas and the way of you talk, really it is an inspire for us how to make the presentation
Thank you so
in
much.
present scenario.
It is my privilege.
So
I welcome if there is
I
any
requested
query.
the participant, if you have any doubts, please ask as a friendly. So as a part of a vote of thanks, on the behalf of the organizing secretaries, I really express my vote of thanks towards Dr. Tiwari sir, who gives immense and outstanding information at a present scenario of antibiotic resistance microorganism and how the AI will support in future in a different views. His presentation, his knowledge is outstanding. I really are thankful to Dr. Tiwari sir, who's spent his time valuable from his busy schedule and comes on the online platform. I thank you to Dr. Tiwari sir.
Thank you so much,
Yeah.
sir.
After 5 minutes,
Thanks.
the validatory function, Dr. A.M. Deshmukh sir joined.
हाँ बन गया सर
yeah. Huh?
Will presided over the Deshmukh sir will presided over the validatory. Okay.
Okay. So
Okay.
you should introduce one.
हाँ.
Yeah.
Okay.
So I requested Dr. Rakesh Thakre sir to take the charge for the validatory function. Over to
Thank
Rakesh
you.
Thakre sir.
Thank you, Dr. Bangiwar sir. Welcome to one and all for the validity function of today's national conference on life sciences, frontiers in life sciences and material sciences at the AI IPR interface. So it was Minds Stroming session started with the Dr. Bhatnagar sir, followed by Mrs. Dr. Pachori ma'am and Dr. Vipransh Tiwari sir. So all the sessions were too excellent and informative regarding AI and IPR application concerning with the life sciences and material sciences. Now we are at the end of 2 days national conference. So we have with us chief guest for today's second day national conference evaluation. Honorable Dr. A.M. Deshmukh sir, Arvind Deshmukh sir, who is the presently president of Microbiology Society India, and he needs no biodata or resume to read because he himself is a legendary character of our Microbiology Society. Everybody he knows him very well, and he is built for the microbiological associations. He provides the umbrella to collaborate with Microbiology Society and conduct such conferences, seminars, and benefit so that the microbiology students all over the globe should get opportunity to interact with the people like today's dignitaries, today's invited top dignitaries. And last day was very fantastic. With we have started with the uh, opening with inaugurated Dr. Um, Akhilesh Pandey sir, who was the former vice chancellor of Vikram University. Uh, we had good speaker, Dr. Vikram Singh Pachlor. He was multi-versatility personality. So actually, we want our students to interact with such people so that they get benefited and learn some new thing which is happening in microbiology, life sciences, and material sciences. So, with this introductory remark, I would like to invite Dr. A.M. Deshmukh sir to please preside over the validity function of today's national conference. Please, Dr. A.M. Deshmukh sir, give your validity remark and conclude the day 2 of national conference. Thank you, sir.
Hello,
Hello. Good afternoon, sir.
good afternoon. Good afternoon. So I hope that you can listen me now.
Yeah, sir.
So at the outset, I thanks organizing committee for calling me again and giving an opportunity to talk in the validatory session. I was present in the inaugural session yesterday, physically. And I appreciated the conference yesterday only. But today, again, there was a virtual session. And the eminent speakers, they have delivered a talk. Your virtual session is equally successful with the offline session. The gathering was a truly national, and the many eminent personality, Dr. Akhilesh Kumar Pandey and one fellow called Chori from the Defense Ministry. They were very eminent. And all principals were present also, Senate members were present. It was a good academic gathering. There are no words to appreciate the team. In the team, all college coordinator, HOD of microbiology were present. And I have seen yesterday, other teachers, their college was not involved, but they were involved. So, it was a good gathering of a young generation. I am calling them young. Why? They are younger than me, no doubt. But they are the young personalities, active personalities in the microbiology. So, as a president of Microbiology Society, it is my duty to give support to all you, because I am getting the fruits. In fact, that was my opinion, that why not in Nagpur? There should be unique example, unique model throughout the India. Like West Bengal, already people are working. In Gujarat, people are working together. No ego, no credit. They are working only for the subject, and you might have experienced, it gives a great satisfaction. Satisfaction is important. I am also getting the satisfaction only. And like me, whenever I see many young people are accompanying me, helping me, supporting me for popularizing the subject, I cannot thank them. But you see, I am very, very happy by seeing your activity. So today is the last day of the 2 days national conference. And definitely it will make a history in the Nagpur area. And I will popularize in Maharashtra also, other state also. You have done the good job, nice job. My support again will be with you, and will do miracle in India on the basis of you, all you. I'm giving assurance that we can do miracle in India. I congratulate you all. I congratulate also attendee, right from England, sir, senior most faculty. Many other teachers were present. I appreciate your efforts, and thanks Thackeray sir and his team for giving me an opportunity to come again in the concludatory session. Thank you. Thank you.
Thank you, sir. Thank you. Now I would like to call Dr. Banira sir for suppose a formal vote of thanks for today's session. Banira sir. Banira sir, are you there?
Only.
असं काही नाही.
आपल्या लोकांसाठी आपण हे सगळे तयार. हे पहा, वाईट
Okay. So as a last part, a vote of thanks. Without vote of thanks, neither any program gets over. And this responsibility given to me by Rakesh Thakre sir. I firstly thanks all the 4 principals, Honorable Dr. G.V. Dadve sir, Principal Yashwantrao Chauhan College, Lakhandur, Honorable Dr. Dahat sir, Principal, Seth Porwal College, Kamthi, Dr. V.V. Hadge sir, Principal Shankarrao Bhoyer Patil Mahavidyalay, Pulgaon, Honorable Dr. Pacharia, Principal Dr. R.G. Bhoyer, Arts, Commerce and Science College, Selu. I really thankful to Honorable Dr. A.M. Deshmukh sir, President Microbiology Society India, Honorable Bhagwanji Khobravgade sir, President Dr. Sachin Bhau Khobravgade sir, Dr. Sanju Kumarji Porwal, President, Honorable Dr. Vijay Kumar Sharma, Secretary, Honorable Dr. R.G. Bhoyer sir, President Shankarrao Bhoyer Patil Krishi Vikas Bahudeshya Sanstha, Warda and Vidya Bharati Sanstha, Selu, Honorable Dr. Abhijit Verulkar, Secretary Shankarrao Bhoyer Patil Krishi Vidya Vikas Bahudeshya Sanstha, Warda. I thanks all the conveners, Dr. Rakesh Thakre sir, Dr. Alok Rai sir, Dr. Ashok Davande sir, Dr. Shilpa Gawande madam, organizing committee, Dr. Raju Gadapaile sir, Suraj Kombe sir, Dr. Shama Murarkar madam, and all the person behind the screen in offline mode as well as online mode. All the presenters give the very well a presentation, their views, their ideas on the AI as well as IPR. It's a wonderful movement. It's a wonderful moment for me to listen mainly yesterday what the AI, what is the role of the IPR. I really thankful on the behalf of organizing secretary to all the delegates, participants, presenters, and a young student. Thank you for sparing his valuable time with us and to make the conference with a great success. Thank you to everyone. Thank you. With the permission of the convener, I close the session right.
Thank you, Dr. Manjunath sir. Now we can leave the meeting. We can end the meeting. Okay?
Yes, sir.
Okay.